we want to make sure that
No,
this gets
thank you.
recorded.
Yeah.
Okay. All good?
Thanks.
Says it's recording now. Okay, cool. All right, thank you for the introduction, and I'll kick us off here. I'll go ahead and share my screen. Some of you California folks may have seen a little bit of this presentation already, so just bear with me, but I'll keep it pretty short. This is just kind of an overview of what has happened so far and what we're doing, sort of where we're at with this insect. So what we know about ecology monitoring and management of Carpophilus beetle in California. You have here Team Carpophilus, myself, Jalindra, David, Sudin, and Rahman. Sort of the first responders, as David mentioned, to these outbreaks when we first started to see them last fall. So a timeline of events in California in 2023, late August, early September, we got a few reports kind of simultaneously from Almond and Pistachio and Madera and Kings County, respectively, about something weird burrowing into the nut. You know, we've been aware of various invasive insects that could potentially affect tree nuts. Carpophilus beetle was on our radar, given that the Australians have been dealing with it for about a decade. And when the photos came across, you know, it was pretty classic signs with the pouty frass and stuff like that. So kind of immediately went out there to look at these things and then do the ID and send it off to CDFA to get the confirmation. And indeed, it came back Carpophilus truncatus. So, you know, that caused us to immediately launch a broader survey of tree nut orchards throughout the San Joaquin Valley. And many thanks to numerous PCAs and growers who started bringing in material. You know, we kind of put the call out, said if you've seen something like this, bring it by Kearney. And so at this point, you know, we've found infested nuts throughout all the counties in the San Joaquin Valley. We found it on almonds and pistachios. We know that it can affect walnuts as well from reports internationally. So at this point, you know, we're seeing it's fairly widespread. The Sacramento Valley, we still need to conduct more thorough surveys through there, and as well as, you know, reevaluate some places in the San Joaquin Valley just to see how far and wide this thing is spread. But the point here is that, you know, we initially had reports on 2 different crops already. Madera and Kings County kind of spread out, and then quickly found that it was, you know, distributed over a much wider area. Given, you know, the awareness we had of this being a major pest in Australia, we pretty quickly connected with David, Madge, and Josh and Ben and all kind of our counterparts out there, you know, to start trying to get up to speed on this thing. And you guys have been, you know, nothing but outstanding in terms of, you know, sharing information, getting us up to speed on this. And, you know, today, hopefully this is kind of a master class with them presenting all their info here on the webinar. So we kind of started building potential collaboration around this insect. And then, of course, you know, the end of the fall, put together a game plan, not just extension materials, but then specific research questions we could start asking, which, you know, the proposal was supported by both the California Pistachio Research Board as well as the Almond Board of California. So very much appreciative of that, and kind of quick turnaround to get money to get this effort going. So here's the beast we're dealing with, Carpophilus truncatus. You may see it referred to as some other species names in earlier literature. That's because the systematics, the taxonomy of it has undergone a lot of updating and revisions and stuff. So Carpophilus beetle, it's a beetle, Coleoptera, it's in the family Nitidulidae. You've got, you know, more than 200 species of Carpophilus globally. The center of origin of this insect is sort of unclear still, but we know in the early 2010s, it was reported on almonds in Australia. In the 2020s, we heard reports of it on walnuts in Argentina and Italy, and then of course the finds in California in 2023. So, you know, this insect appears to be spreading globally. And one of the first articles that came up, you know, when I typed this thing in, the spread of Carpophilus truncatus is on the razor's edge between an outbreak and a pest invasion. So just talking about kind of this global spread that folks are seeing. So in California, we're no strangers to this group of insects. You may have heard of dried fruit beetle, Carpophilus hemipterus. It's a major pest of stone fruit and figs, or the confused sap beetle, another species of Carpophilus. They look different. They don't attack tree nuts, but just to say, we do have related species here in the state that are even on the UCIPM website as pest of other crops, but not tree nuts specifically. What's really unique about truncatus is that it directly attacks those developing kernels. You don't see that from these other species of Carpophilus that we have, at least so far, what we've observed. So here's truncatus. Like I said, the only species that feeds directly on the kernel. These are some photos from the Australian showing the different life stages, the eggs, the larvae, and then the adults. And you can see these photos on the surface of an almond, a kernel, so you get kind of an idea of the relative size. The adults will go lay their egg on the developing new crop nut. They can actually chew a hole through the shell and then deposit the eggs there. Those larvae that come out will tunnel into that developing kernel. And as they do that, it produces this kind of white, powdery, fine powdery substance. It's some mixture of their excrement and chewed up almond material. So you'll see a nut with that kind of fluffy white thing on it, white stuff on it. You can blow that off and you'll see sort of an oval shaped entry hole here. And then, some other images down below. When you cut the nut and look inside, you see this tunneling. The oval shape is pretty specific to this insect. So just some diagnostic characteristics. And here's a better image from Gelindra of one of those shells with a hole chewed on it. So this pest, they overwinter in the remnant mummy nuts. They're thought to have multiple generations per year. We don't know how many. It's one of the questions we're trying to get after, but as said, the larvae will feed directly on those developing nuts. So they're overwintering on those mummies. In the spring, they can become active, and so far what we've seen is they seem to be reproducing on those mummy nuts. But then at whole split, that's when you'll see them not en masse, but some fraction of that population down on the orchard floor on those mummies move up and lay their eggs on and infest those developing new crop nuts. So just some of these photos. Here's that kind of classic thing: the white powder, blow it off. You got a little tunnel there, and here's some photos from Gelindra just showing kind of the relative size of these things on a almond. So we don't really have very good options for monitoring at this point, and I think everything I'm saying here, the Australians are going to, in one way or the other, get way deeper into it. But we don't have a commercially available trap and lure. So the best thing we can recommend is to directly inspect remnant nuts, mummy nuts, or the new crop nuts, especially at harvest. Biocontrol, what we know is very limited. Chemical control, the information we have to date, shows that it's highly variable, primarily due to coverage issues. You have these larvae tunneled inside of the nut. It's really hard to get coverage there. And again, our colleagues, I think, will speak more directly to this, but have tried various approaches to kind of improve that coverage, and still a lot of challenges there. So given that situation, crop sanitation really is critical, and that is the primary strategy that's been used in Australia. And this photo on the right is from one of the studies the Australians did, where they actually took infested nuts and put them at different depths in the soil. And I think in the most extreme treatment, 90 centimeters, so almost 3 feet down, they buried those nuts, and the Carpophilus beetles were able to survive and emerge up onto the soil surface. So I emphasize that because it's not simply a matter of getting the nuts out of the canopy and onto the soil surface, but you really have to shred those nuts. You may need to make multiple passes to make sure they're totally fragmented and destroyed. So don't just let them sit down there on the ground. And to wrap up, just talking about what some of our next steps are going to be, we've put our proposal that has been funded by the commodity boards is to develop a more cost-effective mass rearing procedure. This helps us just get more of the bugs in hand to do studies on. We want to characterize the developmental biology and life history. So what are their upper and lower temperature thresholds? How many degree days that they require to complete a generation? So that can help us with some of the phenology modeling. We want to start working with some candidate pheromone lures that the Australians have developed. I think, again, they'll talk more in depth about that. We are actively trying to characterize the seasonal phenology in California, so monitoring multiple sites across the Central Valley, using the pheromone lures that we can get access to for these research purposes, but like I said, not yet commercially available. So just to understand kind of the timing of these population rises and population growth and development over the course of the year. Determine the spring emergence timing. Evaluate chemical controls. We know there's challenges, but we do have some additional AIs and other methods that Jalindra primarily will be leading that effort to test out. We want to confirm the distribution of the tree canopy. There's evidence from Australia that you have differences in the infestation between the upper and lower parts of the canopy. So we want to confirm that here and also look at that in pistachio. And then, of course, the final objective here, extension and outreach. So online, written materials, video materials, identification guides, how to differentiate damage of Carpophilus from, ants versus navel orangeworm, and hopefully eventually like a grower-friendly ID guide that they can use out in the field, because you're gonna, go out there, look for nuts, and there's a lot of different tiny dark beetles that may or may not be Carpophilus truncatus. So trying to give better grower-friendly support for that. So just with that, want to acknowledge, our lab crew that was able to jump on this survey in the middle of harvest and all the other activities we were doing with research at that time, as well as growers and PCAs who brought us just a mountain of samples from across the valley. So we were able to get a really wide survey, as well as Team Carpophilus here on the call, who also similarly helped kind of extend all this survey effort. So you've got my contact info there. Happy to answer questions here, but I think with that, we'll kind of turn it back over to you folks.
Yeah, thanks, Houston. So far in the chat, a lot of questions about will the webinar be recorded and where it's going to live. And I'll communicate that to all the attendees once we designate where it's going to go. Just had a comment from someone saying that Great Lakes IPM has lures. You want to address that? Anyone want to address that?
Yeah, I can speak to that. So it's not Carpophilus truncatus. It's a lure for another species of insect. So that's not a lure for this. It's kind of deceptively placed on their website, C. truncatus, but as far as we understand, that is not for this insect.
Okay, another question. I see you're evaluating chemical controls. Why not biological controls?
You want me to answer that live? We don't really have a good handle on what potential biocontrols could be. There's kind of a broader project we're going to be putting together here over the summer for 2025 and beyond that, has space for more objectives than what we're currently funded for. So I think biocontrol could fit in there if we determine that as a route we want to pursue.
Okay, that sounds good. There are any other questions that pop up really quickly, we can address those. Any comments, anyone or anything before we move on to the next speaker? All right, well, we're ahead of our schedule, but Josh, if you'd like to go ahead, feel free to go ahead and start.
You got me there, Katie?
Yeah, looks good.
All good to go. All right. So as I mentioned in my introduction, I'm Josh Fielke. So I work for the peak industry body for the almond industry in Australia. The Almond Board of Australia. Today I'm going to take you on a bit of a journey, I guess, and where we've been in Australia and over the time. So I'll go over a timeline of events. I'll hint at a lot of the projects that were funded, which David will talk about, and I'll let him to delve into the findings of those. And then I'll just go over the where to from here. But to start, I'll just give you a bit of perspective on our industry. So our industry effectively follows the Murray-Darling Basin, which is our main water source for the industry. You can see within this circle, that's effectively the river going across there. We got four main growing regions. So we got Adelaide Plains, where probably the first almonds generally were planted in Australia. Then we go up to the Riverland, Sunraysia, and the Riverina. So overall, we have approximately, we'll be just over 62,000 hectares now, which is around 153 acres, 153,000 acres. And we've also got a fifth region, which is called the Swan, over in Western Australia, which is approximately 2,500 kilometres away. So yeah, quite a distance for that grower over there. For context, to put it in relation to California, Stanislaus County is approximately 62 miles wide, and you have 75,288 hectares there. And when you're looking at in Australia, you remember the map I had before, it's effectively a part of our Sunraysia region, which remember is around about the, yeah, it's around the 20,000 hectare mark there, but we'll go into that a bit later. So getting into the Carpophilus story in Australia here, I'm going to have a few QR codes here, which I'll refer to in some reports, and you can go to the page numbers. So yeah, if you want to have a scanner ready, if you want further references, yeah, please do that now. All right, so we delve back when the skies were blue in 2011. Carrot moth was one of our only pests, a major pest of the industry. There was a carrot moth project which was funded in that year, and effectively they were collecting resources around that time, data points, and looking at life cycles and management, and like any other integrated pest management project that was contracted around there. So at the time, in 2011, when that project was going, we had 30,000 hectares in total as an industry. And as I mentioned before, it's about 20,000 hectares that was in Sunraysia when we first saw the pest in the Sunraysia region, which is about, yeah, 65% of the industry there. And you can see the breakdown in the regions there of the distribution has kind of changed now a bit. But yeah, you can see that there, and that's from our statistics booklet, Almond Insights from 2010, 2011. So in just a note here, you'll see I refer to April. It's not necessarily happening in April, but we harvest our product through February up through that period. So that's when our harvest period starts. And I've just used April as a reference because they obviously found these post-harvest. So in 2012, we found unknown pest damage, which was observed as a part of the carrot moth project, and they were going, hang on, this isn't carrot moth damage. So with that, there was no specimens present for identification at this phase. So we had to wait until another year until we get to the next season. So this is what they would have seen. We saw in Houston's presentation before, just the bits of damage that they can see. So there's that QR code there if you want to have a look at a resource. So the difference between Carpophilus Beetle and carrot moth. Probably actually refer back to exactly what carrot moth is. I guess it's just a less aggressive version of your navel orange worm. We don't have navel orange worm here in Australia. But yeah, our pest is carrot moth. Which is quite similar in its damage in the kernel. So in 2013, damaged kernels were collected, and 7% Carpophilus damage was observed at the same site where the samples were taken from in the previous year. It was identified as Carpophilus Beetle Davidsoni, and this image in the picture here is what it looks like at the Davidsoni species. In 2014, mass trapping was conducted as you'd expect. We've got Carpophilus Beetle, let's put Carpophilus Beetle traps out. Similarly, we had that pest in stone fruit. So then, yeah, they went and captured those and obviously took that, collected samples. And they found that that the species that was actually doing the damage was Carpophilus nitidulidae. So we didn't actually know at that point that truncatus was the species. They didn't have I suppose, the identification to be able to do that at that point. But they knew it wasn't, David Taylor wasn't the Mediatus, so they called it Nitidulidae because it was about that. It had similar looks to it. So at that point is when the search started to find the actual species name. So this was a part of the project, still all part of the Carpophilus project. It kind of just took another path. So one of the key points that was found during this time was the effect of water. You can see here when this was an irrigation deficit trial, which you had up from a total of 120% annual water application down to 55% deficit. You're looking there at the different levels of Carpophilus beetle, and you can see the more water that was applied, the more beetle that was present. So it was probably our first key point in knowing what happened to the Carpophilus beetle and where it would like to stay. So potentially in the lower areas, just where water's laying around, all that sort of information. So in response to that, there was 13 field walks held by the extension team and researchers just around the industry to be able to notify them and talk about what's being observed, what management practices to undertake, what we know, something similar to this. So when Houston mentioned before about, yeah, we saw Carpophilus beetle and identified it as truncatus, that would have been nice. It's yeah, I suppose from first seeing it, it's 3 years in the process. So yeah, it took a little bit of time, but yeah, we're obviously getting there, and yeah, we'll go into the next project that was contracted. So really we wanted to learn from that point about the scope and the elements of control for the Carpophilus beetle, what we know, what we can do, how do these traps work, all of that information. So that started in 2015. What we found was through all the trapping that we're doing, the traps weren't sufficiently effective. Effectively, we were seeing damage levels which were high, and then we were getting low trap counts in that area. So while there was some of the species in it, it wasn't lining up. It wasn't making sense. So obviously something was going on, and it was a method that needed to be looked at that point in time. At this moment was when so our biofriendly permit was granted for Australian growers. Our chemical toolkit here in Australia isn't anywhere near, I suppose, have as many options as you do in California. There's a number of reasons for that, but yeah, a permit in Australia is just yeah, it's effectively, we've got this problem, we need a solution now. What can you give us effectively? And we work with chemical companies to be able to get a permit for that. So the issue we're seeing with chemical control options is, as Houston alluded to, was just about able to hit the target. You can see beetles come in at the whole split stage, and they like to hide in the hole. So these holes, these almonds, they face all directions, and your spray is shooting up, and it's really hard to get in. So pretty well what we're seeing at the minute, it's most effective when you've got the most amount of beetle there. So yeah, it's never going to kill them all. Well, it would kill some. So effectively, the solution regarding a silver bullet wasn't that. At that point, I think we were looking for something that was an easy fix, but truly it was at this point we started to realize, yeah, it has to be a completely integrated approach using all different elements of control. And what that is what we still had to find out. So David will probably talk about this in his presentation a bit more, but there was an overwintering study just to confirm where it will, I guess, obviously how it survives the winter here in Australia. So that was conducted over 2016 and started there. And then in December 2016, we worked out that we need a new trap. So they were doing species identification and everything around there. So this is the black trap that was used in stone fruit, and you can see that we're getting a lot of hermipterous And then you can see it was just on 3% there for Nitidulidae. So it was quite an important step, I think, in the realization process in, yeah, okay, we've got all these traps, but what does it mean? We still don't know. So this is at this point where the project was up for a new one, which was then contracted in 2017, and we're looking at different methods for control. So effectively, what do we know, what works, and where do we go from there? So in 2018, it became clear that hygiene is really, really important. I went to California the year prior to this, and hearing David Haviland talk about the levels of control, getting it down to 2 nuts per tree for navel orange worm was something that is to focus on to keep your damage levels low. While those thresholds probably aren't the same for Carpophilus Beetle, and we don't exactly know those thresholds, it's probably a key point is you need to get those numbers real low to be able to control the pest by managing its environment. So through this period, we held a number of field days throughout the regions, just talking about hygiene and how we can go about best controlling the pest and keeping those damage levels as low as possible when obviously we're up against it there with our options. And there's David Haviland presenting at our conference in 2018, and appreciated him coming across for that. In April 2012, sorry, I didn't change that. In 2019, we identified it as truncatus. That's where we found out what the beetle was. At that point, I think we pretty well had a lot of information in what was going on in terms of management practices and what was working and what wasn't. The species name at that point was just, okay, now we know what it is. It helps out the research and everything, and able to work from there. So again, David will talk through this in what he found, but effectively what we found through this project was a lot of bad news, I guess. So if you're going to control the beetle, obviously the burial, if you got mummy nuts on the ground and you want to bury them, that's not an answer. The destruction trials, so if you've got kernel on the ground there and you want to I guess leave it there if you're just checking off the tree, if you think that's a good hygiene job. It wasn't the case. Effectively, the project showed that you have to absolutely shred it to be able to make that not a resource for the Carpophilus beetle. So that was a lot of I guess information there that we had to be aware of, and it pretty well gave us a bit of realization that what we need to do, we need to do a lot more of and put a bit more resources behind it just to be able to, I suppose, control the environment and control the numbers of the pest. So where we sit now in terms of what we're doing, so obviously Agriculture Victoria has continued investment in this new project that they're currently working on and are making progress with the new trap that they're talking about. Also looking at a number of elements to just play a part in that integrated pest management role. As we know, there is no silver bullet in this in pest management, especially when your beetle likes to dig into those kernels and hide in the holes and do everything like that. So the role of variety selection and breeding, I certainly think it's taking a part in our growers in Australia's consideration in what they do in terms of variety selection and orchard design. It's clear that I guess if you're a beetle and you've got an option, how hard do I want to work for my food? Nonpareil, it's a nice paper shell. You don't even have to dig through the shell to get in. So if you have a harder shell there, it can make that a bit more of a challenge and can have a positive effect on your pest damage levels. So we're starting to see that in Australia. So yeah, obviously growers have identified what they need to do, and the two bends will go through exactly what they've seen, both on a grower scale and a processor scale, for different control strategies, and using this knowledge that we've had since 2011 to be able to develop our knowledge and systems around this to keep our pest damage low. And we certainly have seen benefits of these strategies pay off. And another key point is we need to, as an industry, remember it's a true area-wide RPM approach. This beetle doesn't know its boundaries. You can see our density there in Sunraysia. So that Stanislaus County there, 75,000 hectares in there. And we're looking at bit over 20,000 hectares, and it's very around the 30 now. So for Sunraysia in that gap, so you're looking at half the density, and it still moves pretty well in that zone. So it is a true work with your neighbors, know their control options and what their damage levels are, and as much as you can work with your neighbors, the better approach that the better chance you'll have at reducing your pest outbreaks in those areas. Well, that's all for me. I'm open to any questions now. Thanks, Katie.
Yeah, thanks, Josh. That was great. We are still a few minutes ahead. So what I'm going to do, I've got to release a quiz for the first hour. So I'm going to go ahead and post that first quiz. And while I'll give that 5 minutes or so, and I think while that is up, we can talk through some of these questions. We had some really good questions come in. I'm going to go back to right as you were starting, Josh. So a question that we got, someone said, I felt like I saw oval entries in the 2023 harvest, but only NOW present. How exacting is the oval entry? Houston answered that one and said, in his experience, it's fairly consistent oval shape, not circular. The next question, and Houston and David have one. If you want to respond to this, you can. Someone asked, what is known about the interaction between Carpophilus truncatus and NOW? Is there competition over the same resources? You know.
Sorry, I responded to that one. We don't, I know, Jalinda, you've been looking at like how many are navel orangeworm infested versus both versus just one. That's an interesting question. It's something we'd like to know more about, but don't have an answer that's clear at this point.
Well, let me just ask this question. Are both found in the same kernel sometimes?
Yes, definitely.
There you go. So, yeah. I mean, so yes, they can cohabitate. Yes, they can be in the same kernel. I mean, obviously, if one eats the entire kernel, there's no food for the other one.
Right.
You know, so I guess technically they could compete with each other, but absolutely they can both attack the same kernel. They can both live there together. They can be happy and they can be friends.
And also, they can attack the separate kernel too, right? So in that case, I think, pretty much what I've found at least is that they're very small in size. The larval size in 10, 12 is easily fit into within a small space within the nut meat. And navel orangeworm, 2-3 navel orangeworm can still survive in that too. So I've seen a lot of those too. But my just a general assessment is that about 33% I've seen in each category. Navel orangeworm about 33%. Carpophilus about 33%, and then rest, it's kind of combined. So those are based on the tree orchard survey we collected last year, based on obviously 1 year data, but that's kind of, and it's approximately, if you talk about average of the tree orchard. But yeah, they definitely cohabit. But also Carpophilus doesn't need a help from navel orangeworm. That's what I want to say.
Yeah. And that reflects what we see in Australia with carrot moth. We get, I can't remember the proportions now, but certainly proportion with only Carpophilus, proportion only with carrot moth, and then there's always proportion with both. They're quite happy together.
Do you see, I guess, does a nut that has carrot moth in it predispose it to having Carpophilus beetle? Like, are they attracted to damaged nuts is another way to phrase the same question.
I don't think we've seen evidence of that other than having an entry hole that may help.
Okay.
Yeah.
All right, cool. Thank you all. Here's another one. Josh, it seems like some of this was in your talk maybe. Is most Carpophilus truncatus found in orchards with flood irrigations, or do you think it matters? That is something I have been observing.
Yeah, so our experience with flood, our industry is 95% of it is drip, and the other 5% is sprinkler, so micro sprinklers. So we don't have flood irrigation here. Effectively, I think what we've found and where we're finding at the moment is yeah, just where it does manage to get wetter. I see Ben, you wanted to talk about Ben Brown, the irrigation trial. I saw it in the notes there. Probably a good chance for you to do that there here, Ben.
Yeah, we can talk about it once I get into my presentation, but as Josh said, we're predominantly drip irrigated here, so it doesn't seem to matter what type of irrigation system you have. Certainly it needs some sort of level of moisture, so we find it near, more present near the drip line where there is some moisture, as opposed to the mid-row where it's quite dry and hot. So it does seem to bias itself towards a little bit of moisture, not too much moisture, but enough to satisfy itself.
There's a neat trial in one of these reports where you drop the David's team dropped 500 kernels around a dripper line and just went and had a look at how that affected it with moist nuts. And certainly, yeah, that if you're getting those moist nuts, it's a greater environment for them.
I guess another question kind of along those lines. Any growing regions of Australia or orchard characteristics other than drier/water stress that Carpophilus seems to avoid?
Really, areas that don't have nuts hanging on the tree, the better hygiene. If you get, so as we're along the river, if you've got birds flying around and eating your mummy nuts, your overwintering nuts, there's obviously nowhere for those pests to able to build their populations and get through that period with no resource. So space, obviously the density of orchards makes that easier. If obviously you space out the resources, it's harder for them to get through as well. Dave, do you know?
I think,
Josh, do you have? Oh, go ahead, David.
I was going to say also from what we've seen over here, some of the orchards areas that we have that are kind of heavily infested are on drip irrigation on really well drained sandy soil, so it's not as if the ground's wet all the time. But in those orchards, we can get a concentration of Carpophilus in some areas where they're lower patches where water tends to accumulate a bit more, and that is probably most likely partly due to environment, but also just that's where you're more likely to have nuts remaining on trees after harvest. And it's nuts, kind of crop residues in the orchard that are really important, and I'll be highlighting that in my talk.
Okay, that seems like
Hey, real quick, do you have? I remember my visit, you mostly had soft shell varieties. Do you grow hard shell varieties in Australia, and are they more or less or the same in susceptibility?
So there's, I guess there's a bit more of a trend for self-fertility, and a lot of the self-fertile varieties have harder shells. Of the new plantings we're looking at there for a stage, it's about 5% of all new plantings, around that number. So it's not a significant number. We're still predominantly nonpareil Carmel, Monterey Peerless, similar to what you've had, but yeah, since you came out in 2018, Dave, yeah, we're looking at anywhere between 5 to 10% of all new plantings are around that self-fertile.
But no Butte, Padre, Mission, something that's actually a hard shell by our standards.
No.
Okay.
No.
We'll have to figure that out.
Well, one thing, David, that we have a sample collected from Butte Padre next to the orchard with the independent, which was heavily infested. And definitely we saw some damage, but it was really, really minimal low. When we compare to the next orchard, which were independent, we had high damage. So definitely it's a trend, goes kind of similar to Nibelungium. Soft shell, so far Sonora, Nonpareil, Monterey, independent, those are the ones that I've seen a lot, and Carmel also saw to a certain degree, but not Butte Pottery too much. But we have seen some, I think it's followed the similar trend like Nibelungium.
Okay, cool. That was a good little interim chat. Thank you all. Yeah, just a reminder, keep posting questions in the Q&A box. And if I can't verbalize everything, the host or the panelists will answer them in the box. So even if something's been answered, make sure you check that answered column as well, because there's some good information in there. So we're gonna go ahead and move on to the next speaker, which is David Madge. David, if you want to go ahead, and it's all yours.
Yep. Okay, thanks, Katie. Is that showing you okay?
Yeah, perfect.
Good. Okay, hi all again. So I'll be, I guess, trying to address a whole lot of different points that we've learnt from a few years of research on Carpophilus beetle in almonds over here, and where we're heading to as far as research goes for kind of getting into solution to the problem. And all the information I'm presenting on is kind of on behalf of an IPM team, and I'll list them all at the very end. But it's certainly not just me, obviously, with any of this sort of work. So I'll be running through a brief introduction on the beast and its status as pest and why we're fussed about it, life cycle ecology, monitoring options, and management, the current approaches that are available to growers right now, and what we're researching to try to get to a better solution, and then pull that together as a summary. And all these learnings are from 3 research projects that we've had. Our first response to Carpophilus, we had an emergency response basically in 2014 at the behest of, well, requests from industry because of high levels of damage occurring in that season. And then we had an official project from 2015, a 3-year project. And then after that, that was management of Carpophilus beetle, which is really just trying to use the stone fruit trap system, which I'll talk about a bit, see whether that worked or not, as Josh mentioned. And then we've had the 5-year IPM phase 1 project, and we're currently in the second phase of that project, another 5-year project. Its focus is on IPM, or IPM generally, but the real focus is on Carpophilus and carrot moth, with Carpophilus being the major one. So almond Carpophilus beetles, they're in a family called Nitidulidae, which is sap beetles, so they feed on sap and rotting vegetation and fruit, that sort of thing generally. We think there are over 280 species known globally, so there's a lot to choose from. We've got quite a few in Australia. The actual number is hard to determine because some identifications that were done 50, 60, 100 years ago, we have doubts about whether they're actually correct or not because of what we've been finding about taxonomy of Carpophilus. There's a lot of mix-up and misidentifications in specimens, especially in the past. So it's hard to put numbers on some of these things. We do know that when we started trapping in almond orchards as part of our first project, we got at least 12 different species of Carpophilus or closely related beasts, some of which are on the screen there. But this one, as has been pointed out before, Carpophilus truncatus is the only one we have found damaging almond kernels to date. Occasionally we find other species that happen to be blow-ins, but no, 99.999% of what we see is Carpophilus truncatus. So luckily we're just dealing with the one species. Museum records show that it's got fairly broad distribution globally. And it has a really long-term presence in Australia, USA, Europe, and elsewhere. And I'm talking 80 to 100 years. So it's been around for a long time, but for some reason, in very recent times, 2012 for us, and then 2015 Argentina, 2019 Italy, and last year for you guys, as in the States, it has decided that it's going to move into almonds or walnuts as a host crop. We don't know yet why it's doing that, whether it's actually been just accidentally moved or carried into production regions, or whether it's always been in those regions and some environmental trigger has caused it to move on to new crops, because that does happen with pests. So it's not really sure, we're not really sure why it has suddenly become a pest in these crops in different countries around the world. My assumption would be that it's some sort of environmental trigger, like unseasonably wet conditions or something like that. But it is interesting that it's been in Australia for over 100 years. It's been in the US for 80 years or so, but it hasn't actually made it into almonds, which it obviously loves, until very recently. And just one thing I wanted to point out there, one of the reports from a California publication was calling it the Australian beetle. Rather than being an Australian beetle that is kind of spread to crops around the world, it's more like a global pest that is spread locally into crops that it's decided it likes feeding on. We're fussed about it, and we have been for some years, because it's quite a damaging pest. When you look at crop loss and management costs associated with either trying to manage the pest in the field or sort it out in the packing shed, the costs are quite significant. And I've got two types of figures here. In industry, some growers in industry do their own monitoring out in the field to see what sort of pest pressure they've got, what sort of damage levels they're suffering. And those figures can range from 0 to 20+% damage by Carpophilus in particular blocks, or 0 to 10% damage across an orchard as an average. The industry average here is quite a bit lower. It's around, I think, currently about 3.5% in the last season. So it sounds low, but on individual farms or individual blocks in farms, the damage levels can be quite high. Our field trial data, when we've done really high sampling levels, as in a lot of nuts being processed, we've found in our research trials 0 to 23% average damage levels across individual trees, and across individual blocks, 3 to 14% damage. When you're looking at those sorts of levels of damage and the usual value of almonds as a crop, we're talking big dollars. It's hard to put actual dollar costs on the total cost for industry, but it's certainly from some years ago, around 2017 or so, it's been in the tens of millions of dollars. As an added bonus, Carpophilus infested kernels have a real off flavour, so you can't really send them off for, clean them up and send them off for processing as chips or meal or anything like that, because it'll taint it with that off flavour. Whereas I'm not sure about navel orange worm, but certainly with carob moth, if you clean the carob moth frass and crap out, those nuts taste fine. Not that I eat many of them, but they don't develop that off flavour. And that off flavour, we think, is due to the growth of yeasts and other microbes that the beetles actually use to make the nuts more useful as a food source for the larvae. The other big threat that we see is concealed damage as a threat to in-shell markets. Obviously, when you're looking at in-shell crop, it's pretty hard to tell whether it's infested or not. So just visual scanners and sorters are not much use there, and other technology is being used now to be able to look at what's going on inside the shell to try to make sure we don't export too much nut that's actually been damaged or is currently infested. So that's been a real issue. So just looking at the life cycle of the beast, the adults themselves only about 2 to 3 millimeters long, usually around 2.5 millimeters, so they are quite small. They can last 9 months or more. Beetles are one of the most successful life forms on Earth. They're very tough and very long-lasting, very resistant to kind of general enemy attacks. So these tiny beetles can last quite a long time. Eggs, we only have estimates of egg laying capacity from laboratory studies, and they certainly can lay several hundred eggs. Each female beetle can lay several hundred eggs, which gives them a fantastic potential for growth out in the field when they're provided with a lot of food. And the eggs typically hatch in 2 to 4 days. Larvae develop over 4 to 14 days. This is all dependent on temperature. And the pupae 12 to 20 days. So the life cycle itself can take as little as 2.5 or up to about 5 weeks, depending on seasonal conditions. So obviously they can run through quite a few generations in each season, which gives them a fantastic, like I said, fantastic potential for building up very large numbers, especially early in the season, leading up to hull split, which is when they can start infesting your new crop. The eggs, and I think some of these points have been mentioned by Josh and Houston, so bear with me. Eggs are laid in new crop nuts basically as soon as split occurs, and that's, I think, the same with navel orange worm and carob moth. So all three pests follow very much the same pattern as far as development throughout the orchard. And damage starts occurring from basically that whole split period. And the same with, again, Nitidulidae and carrot moth. Damage will keep developing and keep growing until that crop is taken off and either processed or fumigated or treated somehow. So early harvest is one of the key things that we try to do over here more and more. When we're looking at that life cycle and trying to work out, well, where are the beetles coming from? So know what the life cycle is, where are they coming from to actually infest the crop in the first place? So we've, in various studies over the last few years, we've hunted for potential infestation sources in leaf litter and bark in native vegetation, which is around almond production areas where that might be overwintering, which they don't do. We haven't found any of that. We had a PhD student looking at the ecology of the beetle, and that included some trapping in quite areas of native vegetation quite distant from almond orchards. And he did find traces of the beetle, so very low beetle numbers. So we feel that rather than being beetles that are dispersed from almonds, it looks like there are very, very low population levels surviving in Australian bushland. But there's no evidence, we don't have any evidence of any sort of seasonal fly into orchards. We also, as part of a fruit fly project, we got them to, they were collecting different types of fruit fly hosts across Victoria in different production regions to see what hosts, including native plants and so on, might be hosting fruit flies. And they checked all of their species, 22 species of those for Carpophilus, for our beetle, found no sign of it there. But if you want to find them, you look in crop residues in the orchards, especially on the orchard floor, but also on the trees. So what we have is really a situation where, because of the inability to remove all crop at harvest time or all nuts during sanitation processes, we're basically maintaining self-sustaining populations of the beetle within the orchards. And if we look at one of our studies where we actually laid down infested nuts in patches in a few orchards in Victoria and South Australia and collected samples of those nuts during the season to see just what was going on in them. We are looking at a range of nuts here. Some are on dry ground, some are on the dripper line, some are on the tree, and some were just natural windfalls compared to the ones that we laid down. And if you just look at that, the brown bars, so these the tall bars, the dark ones, it gives you an idea of what the adult numbers are doing during the seasons. Effectively, they're maintained in very low numbers during the winter period and through spring, but kind of mid-spring, they start building up and they're peaking around the whole split period here, which we typically is our first week of January, and then gradually die down, relatively low levels, start building up for the next whole split period. Similar to what carrot moth does, where it has a population peak just at the time of whole split, which is very opportune for it. Larvae are focused mostly during mid-spring to mid-autumn during the season. And the one thing that we know from our studies, what I was saying before about sources of beetle, where we kind of looked at different areas where they might be coming from, and the studies we've done, nuts on the ground, nuts on the trees, crop residues is basically the driver of Carpophilus truncatus populations in orchards. And when you look at some of the numbers that you can find in either fresh nuts that are recently infested or old nuts that are lying around in the orchard, you can see how we can build up really large populations by having a reasonable number of nuts lying around in the orchard. I think our largest number of larvae that we found in nuts during some of the early work, where we're looking at infestation of the new crop, maximum I think was 37 larvae in a single kernel. So you don't need too many of those before you get a reasonable number of beetles around per hectare or per acre. And then when you look at the egg production potential, you've got massive potential for infestation. You've already seen these photos. I'm just going to talk about monitoring in a minute. It's really important that we are able to identify which beast we're looking at. It's easy to look at nuts that are kind of chewed and brass and so on and think, okay, it's carrot moth because we've always had carrot moth. But when we look closer, as Houston pointed out, they're very fine powder and frass compared to lumpy, chunky and webbed frass from carrot moth and navel orange worm, and slightly oval tunnels compared to round tunnels for the moth versus the beetle. These are just a couple of the key points for differentiating damage. And it's important for growers who are maybe going out and starting to look in orchards to see whether you have the beetle, and if so, where and how much. It's important to use the resources that are available now to make sure you are identifying the right beast, because especially with an IPM type approach, it's hard to tackle a pest unless you know exactly which one it is. So as long as we can identify what we're looking at, we can go out and do some monitoring. Monitoring is one of the key, I guess, things that underpin good IPM. If we can monitor and work out where we have the pest present, where the hotspots might be, what it's doing during the season, we have a better kind of understanding of its life cycle and so on. We can actually try to develop good management protocols. So when we look at nuts in trees during winter, the mummy nuts, we just call them mummies. High in the tree, low in the tree, generally we can find Carpophilus everywhere, from the very top to the bottom of the trees, but at relatively low levels as far as the number of nuts that are infested. They prefer nuts on the ground. And that's presumably just an environmental sort of thing. When we've actually looked at nuts on trees in the ground to compare their moisture content, nuts on the ground were about 9% higher in moisture content. That may make the difference between them preferring nuts on the ground compared to on the tree. Or it may simply be that they like kind of a lower position where it's less exposed to environmental conditions, birds, predation, and so on. At harvest time, again, we've partitioned trees into top third, middle third, and lower third of canopy, and looked at what sort of infestation levels we have there with Carpophilus beetle. And again, it's not so marked as the mummies in winter, but a higher proportion of nuts lower in the tree have more damage. This is important to know, because if you're sampling nuts off the tree to work out where you have infestation or what the level of infestation is, you'll be overestimating your overall damage level. So we always take nuts off the ground straight after the trees have been shaking, so that we get an overall picture of kind of average damage levels or infestation levels across the whole tree by doing that. We also have now a trap with a lure which is very specific to Carpophilus truncatus, and I'll go through that in more detail a bit later on when I talk about the research side of things. But we'll be able to start using a trap for monitoring as well, and give us much more useful information about population trends, distribution, and so on. And that's what we want to try to get over to California to use as part of your response and management of Carpophilus here. And by the way, I'm sitting in Los Angeles right now, so that's what I'm saying here. When we're looking at management options, I've broken this into a couple of things. One is just management options that are currently available now for growers, and that's really limited to chemical pesticides and sanitation, and harvest as the first sanitation shake, I guess. And so I'll cover those, and then we'll look at options which are new or newly developed or currently being researched, which covers the trapping, biocontrol, variety selection, and post-harvest disinfestation. Chemical pesticides, from my perspective, are a bit of a hairy issue. We have bifenthrin, as Josh mentioned, bifenthrin's available by permit. It was an emergency response, basically, to the appearance of Carpophilus in Australia. We also have clothianidin, which is samurai, and tetraniliprole, Biago. Both of those are registered for use against Carpophilus in almonds. My perception of pesticide use against this pest, as it is with some other pests like carrot moth, is that you may have a pesticide that kills the pest quite efficiently if you can get it there, if you can get it to the pest. These pests are really cryptic, meaning they're usually hidden away inside a kernel, which might be inside an intact shell, which is inside a hull. And as Josh mentioned, trying to get a spray into the split of a hull and hoping that some will get in, past the shell into the kernel to the beetle and killing it, that is a really big ask for any chemical, no matter how strong it is. So even though there may be chemistries and new chemistries which are softer from an IPM point of view, and I point out here with the 3 chemistries that growers can use in Australia, they're either not compatible with IPM, or the recommendation is that they are used in a program that focuses on orchard hygiene. And I'll come to that a bit more later. So the biggest issue with the chemistries, no matter what they are, is I think the delivery of them to where they are required. If you want to protect a newly split nut, as in whole split, you want to protect it from infestation, there's no use spraying the back of it with an effective pesticide. You need to get it into the split. And studies that were commissioned by the the Almond Board of Australia, and studies that have been carried on in the Californian industry, have shown very clearly that getting pesticide delivery to the right spot on almond trees is really, really difficult. I think the California studies are showing that it's 1% or less than 1% of what's in the spray vat actually gets to where it's needed, which to my view seems like not a really cost-effective way of trying to tackle a pest, let alone the side effects where, for example, with bifenthrin and clothianidin, we have observed really serious mite issues. So we start spraying those, we need to start incorporating miticides, and the impact that we're having on any sort of biocontrol of pest mites, for example, out in the orchard, or carob moth or other potential pests, the impacts on those is something we really need to consider carefully before we kind of go down the pesticide track. Having said that, right now, apart from hygiene or sanitation, pesticides are about the only other thing that is currently available to growers. Industry feedback, but the industry guys might have more to say on this, but industry feedback to me has been that there are issues regarding efficacy of pesticide application, kind of, which is what I was saying a minute ago. And maybe I'll leverage the industry guys to maybe add more to that on their view of what they've observed when they've actually seen pesticides being applied for this beast. I do need to acknowledge, though, that the right pesticide can have a place in IPM, but it needs to be the right one that's not really going to disrupt current biocontrol systems, which are often in place for mites, for example. But it also does need to be delivered. And if we can't deliver the right pesticide to the crop, then we have to query whether we should be trying to spray it in the first place, because it may just be wasted effort. So sanitation. We did quite a bit of work on a particular block where we surveyed infestation of nuts, of mummy nuts in winter, and damage in the new crop at harvest, and looked at spatial distribution of infestation and damage and what happened from one season to the next. And I just wanted to pull out one point out of that, and that is that there's a relatively localized link between mummy nuts in winter and damage of the new crop the following harvest time. And it's a relatively short distance thing. It's 100 meters or so, which is what, 100 yards, round about that. So it's not very, very wide distance. It gives us an indication that if the Carpophilus beetle is in an area because there's a concentration of crop residue in an area, they tend to be quite happy staying in that area. There's no reason for them to kind of fly off to anywhere else. They have a food resource, why move? And whether we look at the number of nuts remaining on trees in winter, or the infestation level of nuts on the ground in winter, infestation with the beetle, both of those independently relate to higher levels of damage at harvest. And they're obviously related. The more nuts you have on the trees in winter, the more nutfall you'll get, more nuts on the ground, so greater resource for the beetle to build up. But independently, those two factors lead to higher levels of damage at harvest. So the point that I try to focus on with our growers in Australia and with anyone looking at the beast in California is that sanitation is critical. It's been critical for a long time with carob moth and your navel orangeworm. It's really critical for this beetle because of its potential for damage. And regardless of what we come up with as far as other management option goes, sanitation is going to be critical to try to keep population levels down to a point where biocontrol or trapping or some other approach can actually have an impact. If we don't have good sanitation, we build up really high population levels, then it is very, very difficult to try to manage that with any sort of control technique. So sanitation, sanitation, sanitation. There are obviously a whole range of different approaches for sanitation, different bits of equipment and so on. Picking up nuts, taking them off farm, selling them for cattle feed, and possibly stockpiling them and treating them, fumigating. The new inside track. I'm not sure exactly where that is right now, but there's potential for that sort of gear of actually targeting mummy removal more effectively than shaping trees, because we have difficulty getting nuts off trees. Pulverizing nuts on site and burial. And I'll come to burial in a tick with a couple of points about that. The main thing is that we get nuts off trees and out of the orchard or destroyed on site. And when we're talking about destroying, we are talking about pulverizing them quite finely. We have found Carpophilus larvae in 1/4-inch almond chips after the area had been kind of shredded with a mulcher, with a kind of a light duty mulcher. So if we're pulverizing the nuts, they need to be quite small and scattered across the orchard floor, not dumped down in some sort of windrow, because that'll just be a nice environment for them to feed and survive. So range of different options. Growers need to obviously choose the option that suits them best. We can't do polling in Australia because of the cost. Our biggest issue is getting nuts off trees. We have quite a different environment, which I think Ben Brown might cover off on, which makes it more difficult for us to actually remove the nuts. Just to look at the burial side of things, and I think Houston touched on this. Burial was tried as an option for disposing of nuts once they'd been kind of shaken and swept out of the orchard. And we did a burial trial where the deepest we buried them was under about a yard of soil. And we monitored that. So we buried them in May and monitored them through the season. We did subsampling of the nuts that we're burying, so we knew about how many beetles we're putting down on the soil. And in spring, we started getting emergence from beetles from the soil from the different depths. From the 2 shallower depths, 30 and 60 centimetres, so 1-2 feet, we ended up with a hell of a lot more beetle coming out than we buried. So what we'd actually provided them with was a really nice, safe, stable environment to keep feeding and keep breeding. So that was obviously not great. We terminated that trial before the deeper, the emergence from the deeper burial depth had actually peaked. But my gut feeling is that that would have carried on and produced a lot more beetles than we buried in the first place too. So if anyone was looking at burial as an option, and burial has its issues, as in cost and inconvenience of the equipment you need and space to dig massive pits and bury, but my gut feeling is it would need to be quite deep, so probably below 5 feet, given what we saw coming out from the 3 feet in the soil. So that's something to bear in mind. Personally, I think burial is not the way to go. When we're talking about sanitation, a couple of key points, whatever sanitation techniques being used, and that's timing and efficacy of the sanitation system. If we look at a couple of the bits of data that we've just seen a few minutes ago, when we were tracing nuts, looking at nuts on the soil surface during the season to see what the beetle infestation levels were and so on, we started to get a build-up of adults in early October, so early to mid-October. And when we buried nuts, we got emergence in around the same sort of time. So it's sort of indicates that you really should get nuts out before that time of the season so that you're not allowing the beetles to start dispersing. Last season, with the new trap that we developed, we had some of those handed out to growers to start using them as monitoring traps to just see how it went. And we actually started detecting significant levels of moth activity in mid-September. So it looks like we actually need to go quite a bit earlier with sanitation if we want to stop beetles from dispersing from the nuts that are actually in the orchard. So ideally, the crop residues in the orchard would be removed by early spring, potentially even earlier. We don't know yet. Having a trap that actually works well, though, will give us an opportunity to start monitoring beetle activity right throughout the season, so right throughout the years, to give us a much better handle on if we're going to perform sanitation or some other management issues, what sort of timing do we really need to tie in with according to what the beetle activity is. That will obviously vary season to season because of different seasonal conditions, temperature and other environmental factors and so on. But with enough trapping data, if we start collecting over the years, that'll give us a fairly good indication of what to expect. And this is one thing that we talked about sanitation a lot in the almond industry. One thing that I don't want people to forget is that the best time to actually remove nuts is obviously at harvest when they're worth money, rather than in winter when they're kind of rubbish to be disposed of. And I won't stop saying that it is something that, even though sanitation may be difficult and harvest may be difficult, it's something that we shouldn't lose sight of. If there's anything at all that we can do to improve harvest, that will benefit us in the long run as far as management of these pests goes. And it's not just Carpophilus Beetle, it's navel orange worm and carrot moth as well. We did a few studies looking at the efficacy of sanitation efforts because we knew that sanitation was critical and we wanted to see exactly how it was working. And in one of these studies, we found that it's hard enough getting nuts off trees in the first place, but when we did get them off, sweeping actually left around 50% of mummy nuts along the tree rows themselves because of soft soil and uneven soil and holes and pits and so on. And that's too much. That's leaving too much nut behind. Once nuts are in a windrow, we're actually quite efficient. So the standard harvest pickup machinery gets up to 100% of the nuts out of the windrows, which is pretty good. The problem for us is that continual nut drop is an issue. We haven't been able to shake all the nuts off in sanitation shakes. So even after we sweep the orchard floor clean, we still have nut drop continuing basically all year round. So it comes back to nuts on the trees being an issue, which comes back to more efficient harvest. The recommendation is basically continue shaking, sweeping and disposing of nuts as long as possible before you start shaking buds off or disrupting the new crop and shaking them off too early. Because the more nut we can get out of the orchard before hull split, the better. As yet, we don't have any sort of threshold for Carpophilus beetle. It's going to be low because of their potential for building up high populations, but the amount of work involved in that, we've steered clear of trying to develop a threshold. We did try very early on, but the trial was wiped out by bird activity because it was a relatively small, isolated property. We're looking more at the actual management and not too worried about thresholds. The lower, the better is our message. So on to trap development and lure development. And when we talk about trapping, we're talking about the physical trap and the lures themselves. There has been a commercial trap available for growers for managing different species of Carpophilus beetle in fresh stone fruits because they can be very damaging in peaches, nectarines, apricots, and so on. So that was developed a few years ago. And it basically is a trap, plastic bucket trap with a funnel inlet, a pheromone plug, little rubber septum, which has pheromone compounds embodied in it. And those compounds are basically aggregation pheromones that the beetles produce. So when different Carpophilus species get to a good food source, it's like getting to a barbecue. They say, hey, come on in, there's food here. So they release a pheromone, which attracts male and females of the species. So that basically it's telling them that's a good food source here because they only produce it when they're feeding. So that pulls beetles into the vicinity of the trap. There's also a tub of liquid, what we call coattractant, which is really plant odors and fungal odors, which the beetles find really attractive. And there's a pesticide strip in their little yellow square, which kills the beetles into the trap. That trap can be used quite successfully in stone fruit to protect crops from damage. And it has been used for some years and it catches the bulk of beetle. When we tried that in almonds, about 3% of that catch was Carpophilus truncatus. Most of it was other species which don't damage almonds. So the task of our first phase of the IPM project was basically to build a better trap. So that project isolated and identified and synthesized the pheromone that Carpophilus truncatus produces, specifically that species of beetle. We also modified the coattractant by pulling out compounds that we think might have been repelling this species of beetle and modifying the blend of what was left. And also identified a more effective trap itself, so the trap body and placement of the trap. Within stone fruit, the traps are typically placed on 4 or 5 foot stakes around the orchard or in the orchard. And we found that placing it on the ground was much more effective. So the trap body itself, the trap placement, pheromone and coattractant were modified to get a trap that actually works for this species. The work behind that was a hell of a lot of very intensive and how to say it? Yeah, very intensive and technologically advanced work went in behind developing the new trap. I'm just showing one little bit of equipment here, which is quite pivotal to the work. For anyone who really needs to know, this is a gas chromatograph chromatograph, mass spectrometry, flame ionization detector, electroantennograph. And basically what it does is allows us to trap a truncatus beetle. And remember, these are only about 2.5 millimeters long, so it's quite fiddly to do this. We can pass an airstream over one of its antennae, and that airstream is one that we've selected that has a range of volatile organic compounds in it, which we think are likely to be attractive to the beetle. And we have a probe which is inserted into what we could call the beetle brain, I guess, to measure electrical responses to what is being passed over the antennae. So basically it's a way of putting compounds over the antennae and measuring how the antennae responds. And to get a sort of readout like this, which is showing a little chart of activity on the antennae itself. And at the same time, the analytical equipment is analyzing that airstream and telling us exactly what compounds, peaks of different compounds are occurring as the airstream goes over the antennae. And this allows our chemical ecologists to look at particular responses from the antennae and what compounds they might relate to. And then with a fair bit more work after that, using a whole lot of laboratory study techniques, looking at attractiveness, it allows them to look at attractiveness of particular compounds or repellency of particular compounds to the beetles using a range of different study techniques. Choice tests, for example, of whether the beetle will go down one tube or another, depending on what odor is coming out. And they can actually work out that the particular compounds that the antennae are referring to, which ones of those are attractive, and then which blends of those or concentrations of those are most attractive to the beetle in the lab, and then turning those into lures that we can get out into the field and put into traps and actually look at, do they work in the field as being effective for trapping the beetle? So 5 years of work using all this sort of technological approach in the lab backed up with field trials in the field have got us to a point where we have a trap which is now on average catching 96% truncatus. So rather than a range of different species, it's very specific to the one that's damaging almonds. We now have in that trap a really good monitoring tool and potentially a management tool. So that was a large focus of our first phase of the IPM project, was simply to get a trap that actually works. And now we're in the phase of trying to make it work as for management for growers. This past season, we handed out a few traps to growers in Victoria to start looking at how they went using it as a monitoring tool and see what data we could get from those traps as far as tracking Carpophilus truncatus activity during the season. This is just an example of one set of data from a grower. And having these traps out in the field will allow us to look at things like relationships between catch of the beetle at different times of the season and how that might relate to damage levels at harvest, to give us the potential for kind of forecasting risk levels. So it gives growers maybe a bit of forewarning about whether they're at high risk of damage and whether they need to really take more activity earlier in the season. It'll also allow us to look at the influence of seasonal conditions on beetle activity. Why do we have high levels of activity or damage, low levels of activity of damage in different seasons? Is it purely due to rainfall? Is it other climatic factors? And it'll also allow us to look at potentially the impacts of management practices that growers are conducting. So from now on, we actually do have a really good management tool, which is a monitoring tool, which is very specific to the damaging species of Carpophilus. And we're looking forward to seeing what data we can actually get in from the grower community to build on our knowledge of the beast. The other angle of trapping is looking at mass trapping. The big trap I showed before, the big black one, is the one that's used successfully in stone fruit for mass trapping of Carpophilus species of damaged stone fruit and actually protecting the crop from damage. What we started last year is looking at using this new trap for Carpophilus truncatus as a mass attract and kill system to see what impact it had. And just to explain briefly, we had in a commercial orchard, we have 5 pairs of plots that we selected to be as similar as they could to each other. And one of those pairs we left without any traps at all. The other one of those pairs of plots, we put traps in quite high numbers, which I'll talk about in a minute. And then we went in and took our harvest samples at the commercial harvest time across those plots and analysed them. And these percentage levels are the percentage reduction in damage across those plots with the trapping regime in place. So an average of just over 56% across the farm, as much as almost 80%. So that looks really promising as a potential management tool for the beetle, not just monitoring. We need to keep in mind, though, that we went in to basically prove whether this approach could work. So we went in with really high trapping density, 16 traps a hectare, or 6.5 per acre, which I feel is obviously more than growers could basically handle to be cost effective. What our role is now in the kind of ongoing work in this project is to look at ways of making attract and kill, or mass attract and kill, cost effective. One of the ways is obviously to look at the effect of lower trapping densities, just to reduce the number of traps and the amount of maintenance that goes on with them. Another angle is to be looking at whether we can use the lure that we've developed, do away with the trap itself, but use the lures that we've developed together with an insecticide of some sort to actually attract beetles in to a small dollop or squirt of spray or something on trees, where we could actually attract and kill them without actually having a physical trap that needs to be maintained. That's all potential work for future. But currently we have a trap that actually works very, very neatly for the species. We just need to try to make it work as a management tool. When it comes to biological control, I said before that beetles are probably the most successful life form on Earth. There's very little in the way of natural enemies, as in other insects or predators and parasites, that tackle Carpophilus beetles. Very, very few. Whereas with, for example, our carob moth, there are dozens, many dozens of potential natural enemies that tackle them. One thing that has been highlighted, though, with some of our work looking at this whole area of biocontrol, is that entomopathogens, which are basically insect pathogen diseases that kill insects, have been highlighted as potential biopesticides for Carpophilus. And these are particularly in the areas species of Bovaria and Mycorrhizum, and there are commercial products available with these species these fungus species to be used as pest control, applied pest controls for particular species of pests. We are currently researching the use of some of these species as controls for Carpophilus truncatus. Some of our early studies have shown more than 90% larval mortality with the beetle, with entomopathogen strains or isolates that have been collected from almond orchards in Australia. And they perform much better than the commercial strains that aren't meant to target Carpophilus or for other species of insect pests. So just that level of mortality is really promising for early studies of entomopathogenic fungi as a biocontrol or a biopesticide. The research it's happening right now. It is ongoing, looking at developing, or the potential for developing, these fungal isolates as biopesticides for application to almond orchards for truncatus control. And we're looking at collaboration with private industry research as part of that work. So there's real promise there, but it's really early days yet on this one. We're just fingers crossed and hoping that something comes out of that, because that would be a very neat way of managing a pest without actually spraying contact insecticides that have a whole lot of issues associated with them. As I said before, this sort of biopesticide would probably be applied through irrigation or a soil application, and given that truncatus prefers nuts on the soil, and it's kind of, you're liking that environment rather than trying to spray them on the tree. But like I said, early days. One thing that I think was mentioned earlier, Josh might have brought it up in a few comments on it, about variety selection. Variety selection, I think, has a lot of promise as a longer term aim for tackling Carpophilus truncatus and potentially other pests. During one of our trials, when we were assessing harvested nuts, looking at some treatment effects to do with sanitation, I went to the trouble of inspecting the nuts, the in-shell nuts, really, really carefully to see whether they were actually split open. I mean, that's obvious when they're split open like that, but also ones that appear to be shut, see whether they appear to actually have opened at some stage and then closed up again, or whether they really were sealed. And when I separated out the ones that were just absolutely sealed, had not split open, and then everything else in another heap. So some nuts that looked as if they were sealed, but I could actually slip a bit of stiff paper in through the suture, so it had opened at some stage. separating those nuts out, the nuts that had never opened compared to the nuts that were unsealed, either open or opened and then closed again, chalk and cheese as far as Carpophilus being in them. Carpophilus does chew through shells, but I've only seen it over here where the nut has just recently gone through the hull split and the shell is still quite white and soft. We've definitely seen Carpophilus damage in those nuts. But once the shell has hardened off a bit, we simply don't seem to find Carpophilus in there. It's as if they are either too lazy or unable to chew through themselves. That's our general feeling. Different varieties, it may vary, I'm not sure, but this is just an observation that I want to put out, that as far as long-term management goes, rather than relying on the silver bullet of a trap or a spray, the soft spray, or continual efforts on sanitation and so on, one way of approaching the pest problem is to say that if slightly harder shells or completely sealed shells actually exclude a large amount of infestation, then that'd be something for the industry to aim for in the longer term, because we really don't want to be coming back in 25 years and talking about the same problem when over that 25-year period, the industry has opportunities to change over to varieties that are not as susceptible. And getting rid of nonpareil, which has a lot of open splits, would get rid of a whole lot of other issues to do with contamination from when they're rolling around on the soil, or bird excrement getting in, or rain getting in and staining shells and so on. So that's just something, it's not something you do overnight, obviously, but it's something that should be in the planning, kind of the planning regime for the almond industry generally, is trying to get away from really super soft shell, split shells, because they're generating a lot of issues for us. Just having a look at post-harvest disinfestation. Part of the IPM project's remit has been looking at improving post-harvest disinfestation, because we know there have been real shortcomings in it. We use phosphine, and the protocol is to maintain at least 300 parts per million in the atmosphere within an almond bulk heap for 7 days to get good kill of the various post-harvest pests. Very few of the commercial fumigations that have been monitored by our post-harvest researcher have got to 300 parts per million. And none of them have certainly got to 300 parts per million for the 7 days. No, we often run out of phosphine in just 2 days. And it turns out that a lot of phosphine is absorbed by the hull and shell. Our research has been looking at developing new protocols for fumigation with phosphine. And it's looking like, rather than trying to go in with a really big dose to start with, that's just absorbed as well, but going in with a standard dose and then going in with a follow-up kind of standard dose gives much better response. As soon as the nuts have been fumigated once, their absorption drops quite markedly. So researchers looking at how to actually treat different varieties of nuts and different maturities of nuts with phosphine effectively. The one bright point that's come out of those studies has been that Carpophilus truncatus, luckily for us, appears to be quite susceptible to phosphine. So killing it with phosphine doesn't seem to be an issue, but actually getting effective phosphine fumigation is the bigger issue. So that's something that's being worked on. So just to summarize all of that, from our experience over here in Australia, Carpophilus truncatus is the only culprit to date as far as almond kernel damage goes. It is a globally distributed species that seems to have taken a liking to nut crops for whatever reason, we're not 100% sure. It can cause and has caused very significant crop loss and costs in Australian almond crops. And as I mentioned before, direct crop loss from kernels being destroyed, and the costs of trying to manage it with pesticides or sanitation and so on, and sorting in the packing shed to get rid of contaminated crop. So quite a lot of cost there. The populations are sustained on farm because of all the residues we leave behind. We leave a fantastic resource for them there in leftover crop. Current pesticides have efficacy and off-target issues, which really make them undesirable. They're certainly used in some cases, but they're not desirable by any means, because especially the off-target problems. Effective harvest and sanitation are likely to remain crucial to management of this pest, and for us, carrot moth as well, and navel orangeworm, regardless of what other management techniques we come along with. If we can't suppress the population growth by good harvest and sanitation, we're going to have a really hard time to manage them with other means. But a bright point, we do have a new trap, which is currently immediately a really good monitoring tool, which we hope to be implementing a lot more widely this coming season for us, and hoping to develop that or a version of it into a viable management tool for this beetle. That's where our fingers are crossed. Entomopathogenic fungi as potential biopesticides are showing a lot of promise, but it's still very early days, so we won't know where they head for a while yet. And just one last point is that if we're looking long-term for the industry, moving away from things like nonpareil that have very high levels of shell split would certainly help kind of reduce the impact of Carpophilus beetle as an issue for industry. And I'll leave it at that, Katie.
Sounds good. Thanks, David. That was great. Super thorough, and I think it's just really helpful. I am going to go ahead and launch our second quiz, and I'll give it 3 or 4 minutes or so. And while that is up, I think if we want to address some more questions, let's see, it looks like a lot of them have been answered, but it's helpful to reiterate. Here's a good one. So provided that ecological factors affect the population dynamics of Carpophilus truncatus, how good enough can we predict the future population from the trends observed with multiple years of previous population and weather data?
Right. We don't have multiple years of population levels. What we do have is damage levels in crops, and some parts of the industry have been maintaining records for a few years, and I'd certainly have access to that and been using that and want to look at predictive capabilities in future. What will be most useful is when we have a lot more monitoring data from the traps, so we can actually see what population trends are and then tie that into damage levels, environmental parameters and so on, or environmental conditions. Just because we have a high level of damage, in a block, and it's very sporadic. In one farm, we can have a block that kind of registered 0 damage, a block that registered average 25% damage, and everything in between. So it's hard to actually tie down what's going on here without having more detailed monitoring data like traps spread across farms.
Mm-hmm.
It's obviously not purely environmental conditions, because we can have very different population levels in a block. It's going to be, I imagine, interaction between environmental conditions, the blocks themselves, whether they have low patches where moisture accumulates and they're more mummy nuts and so on. So there are quite a few different factors there to tie into what the risk is going to be as far as kind of damage at the end of the season, or even just population trends in the beetle. We're not there yet, but we hopefully are collecting a lot more data that will be useful for that sort of thing now that we have a monitoring tool. Because monitoring, going out and doing the level of surveying of nuts and level of surveying of harvest damage levels that's required to get some of that data is a huge job. Traps make it much easier.
Yeah. Okay.
Yeah.
Another one that seems interesting. How much might off the ground harvest help? Obviously, you'll still have mummies to contend with, but any thoughts on that?
So I'd be interested to hear from the others too, but from my point, they are looking at off the ground, basically off-ground harvest over here. One of the biggest issues is that all of the varieties that I know of have quite a bit of drop before you can go in and shake the tree and do a shake and catch. So taking account of the nuts that are dropping off early is an issue for them. So they still have to do some pickup there. The biggest issue that I can see is that when we grab a tree by the trunk and shake it like mad, it simply doesn't get the nuts off. So whether you shake it onto the ground or into a catch system, the current setup and the current tree size and so on just typically doesn't allow us to get all of the nuts off the tree. And as soon as we start leaving a few nuts on the tree, we start having these issues. Now, I know the Californian industry developed the 2 nut per tree in winter as a threshold some years ago. When we have sampled blocks in the past few years, it's not unusual to find trees with 200 nuts or more, and an average, a block average of maybe 80 per tree. So it's a really different situation. If you guys are telling the truth and you do get down to two nuts per tree, we've got a very different situation over in Australia because partly the environmental conditions that I think Ben will talk about. So yeah, whatever the harvest system is, you actually need to get the nuts loose off the tree in the first place. That's the issue, whether it's shake and catch or off the ground.
David, what would the effect of when I think about off ground harvesting, what would be the effect of, like, you're still getting the pest there through hull split? Would that be significant? Like, how would you see the reduction?
Sorry, what was that?
So you're getting the pest coming, so Carpophilus enters that hull split.
Yeah.
Would off ground harvest change the result that much?
Look, if you could somehow, yeah, probably not. If you could somehow shake and catch all the nuts off a tree, then you're done.
Yeah.
My understanding is that you can go and shake and catch before they hit the ground, but you've lost the early part of the crop that's already gone on the ground. And if you're leaving nuts on the tree, then they're still going to fall to the ground anyway, and Carpophilus is still going to have a field day with them. What the Australian industry has done a bit is looking at earlier shake, so going to greener nuts. The idea would be to shake nuts off the tree and actually get them off before hull split, but there's a limit to how early you can go and still have the kernel mature enough to actually end up being a really good quality kernel. So earlier shake reduces, earlier harvest will reduce damage simply because the longer nuts are on the tree, the higher the levels of damage or infestation with any of these pests. But you're right, Josh. Given that these beasts are getting in at hull split, I guess the issue is really getting nuts out of the orchard. It doesn't matter how you do it. Like if you can shake every nut onto the ground and then pick them all up, no, that's the answer. You don't have to do shake and catch. Some of the more intensive growing systems that the industries are looking at, like really tightly planted windrow or hedgerow type trees, they may be more amenable to some sort of harvest system, which is not necessarily shake and catch, but some system which can actually knock every nut off. And if we ended up in a position like that, then we'd actually be getting somewhere as far as harvest goes, and there'd be no need for sanitation, really. That's the key thing, is getting the crop off in the first place.
Jalindra and David, you can chime in, and then we're going to move on to Ben after you two.
Oh, I actually have a question for David. So far, what we have observed in the mummy nuts, we never seen the pupa on those mummy nuts, so we're assuming that, or based on some of the other literature, they are pupating in the soil. But most importantly for me is when the hull split happens. After that, when the beetle come up and they start laying eggs on the hull split nut, how many generations after that you expect they will complete? And because we've seen larvae as well as the adults inside those nuts in there, we never see the pupa. So in that case, do you expect those larvae feed on and become advanced stage and drop on the ground and pupate, and again, adult will come back and do the couple of generations? Or is it something, just one time after the hull split? Because that would have a lot of implication in terms of, late variety, early variety, all of that, and how much damage you expect.
Yeah. So at that time of season for us, they'll be running through generations pretty quickly, like 2.5 weeks. So given that we start hull split, let's say the first week in January, we start shaking. I mean, typically the second week of Feb, some people are going in a bit earlier. So that's 4 weeks already. Harvest can go through, and again, the bends might cover this a bit more, but harvest can go through to March, April, May. In 1 year, I was doing some sampling and harvesting was still happening in June because of wet weather that delayed it. So you think if you leave a couple of months after split starts, or 10 weeks after split starts, you've potentially gone through 4 generations. So and they're going to keep ramping through because there's a fantastic food source there that nothing's going to stop them until you fumigate that crop or process it. As far as the pupae goes, yeah, that's one thing I didn't mention. We find very few pupae, and we simply haven't had time or haven't made time to do the work to do a lot of digging to see where they are. My assumption based on Carpophilus knowledge generally is that they'll be pupating in the soil, which is why we don't find pupae and nuts on the soil when we're monitoring those closely. And yeah, you'll have, I think we've found one or two pupae in nuts, but they were nuts in captivity where they couldn't actually get to the soil in the first place. So they'll be dropping from the nuts, either that are lying on the ground or on the tree, pupating the soil, emerging as adults, flying up, infesting new crop. And remember that they'll also be like carob moth, and I assume navel orangeworm. Not every beetle is going to rush off to the new crop nuts and infest them. Otherwise, we'd be harvesting nothing. They still love mummy nuts because the mummy nuts are pretty grotty. They're infected with yeasts and molds and all sorts of stuff. That's really attractive to the beetles. So they'll still be cycling through crop residues that have been sitting around for a long time, and a proportion of them will go to the new crops. And any beetles emerging from the soil, if there are nuts nearby, then they're probably more likely to go for those because that nuts are probably more attractive than flying off up to the trees and trying to find a nut that's split and that's at the right stage. So did that answer your question?
Yeah, thank you.
Okay.
Yes, yeah. So David, I don't want to steal Ben's thunder. So if I'm jumping on his topic, just let's let Ben answer it. But one thing that hasn't been done on this call is to sort of contrast California versus Australia from a production practice standpoint. So for example, you have very shallow soils, very fibrous roots, you're watering all the time, very high humidity, which may be why an entomopathogen works well for you and may not for us. We don't know that. Likewise, that really high humidity you generate, it causes a lot of hull rot. Hull rot generates a lot of stictites, which is why I think you have a lot more difficult time getting mummies out of the tree than we do. We tend to have a lot more removal, a lot easier because we have much less hull rot, which of course leads to sanitation costs, which of course, all those things are tied together. So I don't know if at some point, whether it's Ben or someone else, it'd be nice to kind of draw those contrasts a little bit because, for example, for us, it's going to be easier for us to sanitize from a standpoint of getting nuts out of the trees. At the same time, all we have is a few crummy crows and people who don't understand what a rainbow lorikeet can do. So you have benefits that we don't have. Yep. So yeah, at some point, if you can kind of address that would help us to put a lot of your comments into context.
Yep. I think, Ben, you're addressing the difference between the two.
I am, David. Yes, and David. Yeah, we'll talk about the contrasting conditions.
Perfect. I will rescind my question and look forward to your answer.
But I will add one thing on there now that you brought it up. It's interesting that the big issue in California seems to be hull rot and stictites because we've wondered about that over home in Australia. And we did do a few studies on that. I didn't go into this because it was for the time side of things. But when I've gone out in the orchard after harvest, if I go out there and I did this a few times with a branch shaker that I borrowed from the Pistachio Growers Association, and most instances, when I went and shook a branch that had a lot of nuts on, they just came off very easily, kind of after the commercial harvest had started. So they weren't actually glued on. And a small trial we did where we stripped every single mummy nut off a series of trees and then looked at what happened from then on as far as nuts left on after the following harvest, hull rot strikes, nuts remaining on, hull rot struck shoots and that sort of stuff. The disease management team, which we're operating in parallel with, an integrated disease management team, in that study, they found no real association between hull rot strikes on those originally clean trees and nut retention on the affected shoots. So I think there's something there that's going on and maybe need more work in Australia on it, but it doesn't seem like you have hull rot, you get nuts stuck on. It doesn't seem to be as clean a story as it is in California, where it seems you get hull rot, you get nuts stuck on. So I think there are a few issues, and there are a whole lot of nutrition and water issues that can stick nuts on as well. So yeah, there's more to look at there about why nuts don't come off at harvest. And if someone can solve that for us, then we're kind of halfway home.
Yeah, thank you. That's great. Yeah, the other thing I saw in Australia is a lot of times there's no nuts in the bottom half of the trees in Australia.
Yeah.
Whereas we have the nuts throughout. And of course, if you're trying to shake the nuts out of the tippy top of a tree versus the bottom of the tree, it's just easier. So yeah, I'll interested in Ben's comments. Thank you.
Right. Thanks. Yeah, that's a perfect setup for you, Ben Brown. Go ahead whenever.
Okay, thanks, Katie. Let me just share the screen.
Perfect, looks good.
Okay. All right, welcome everyone. Yeah, pleasure to be here. Thanks for the opportunity, Katie and others for inviting us along. So as my presentation will be from a context and a lens of production and growing. As I said, we farm about 22,000 odd acres here in Australia. So that'll be the basis, I guess, of our presentation here. I've got about 20 slides, so we'll try and stick to the time limit. Okay. All right, so Josh gave you a bit of a background, I guess, as to where we grow the almonds in Australia, so I won't labour that point, but basically we're here in the Southern Basin, and the whole picture of what you see here is the Murray-Darling Basin. So it's a confluence of various rivers, ultimately leading into the Darling or the Murray, and then ultimately the Murray, which goes out to the sea just near Adelaide. Today I'm in Renmark. You can see here on the map here. So, but predominantly the almonds are farmed in the southern area. Now, I guess I'll just make why I draw this to your attention is later you'll see some maps around rainfall. So, but if you just picture this map and where we're located, so we're located between Renmark, Mildura, Swan Hill, Griffith. That's the sort of region there that the almonds are grown. So, to David's questions and the contrasting conditions between Australia and California. So that's the first few slides, to be honest, because we are very much the same but different, which is a bit of an oxymoron, but there are very similar conditions between the two industries and two areas, but there are some very clear distinctions as well. So I guess firstly on the soil front, we predominantly, as you can see there, approximately 75% of the Australian industry is farmed on what we call the highland mallee soil. So they're typically lighter in texture. They vary in topsoil depth from 20 to 30 centimetres of topsoil. Those topsoil textures would be sort of loamy sand through to sandy clay loams, with generally some sort of neutral to alkaline in nature. And about 25% of the industry is located on broadly floodplain soils of some description. As you can see there in the box on the left-hand side, that sort of is capturing the topsoil in the various layers, carbonate layers, the limestone, which can sometimes be sheet limestone, and then what we call the Blanchtown clay. So in Australia, in this southern area, we have this impermeable layer in 75% of the industry, which is called the Blanchtown clay. Many millions of years ago, that this part of the country was an inland lake. And it was that clay layer that allowed this area to become an inland lake. So that is the origins for lots of our troubles in this area when we come to irrigated horticulture in the broader sense. And that, coupled with the variability in the topsoil depth and the variability in the elevation, as you can see there, we are largely a landscape in this area. So I'm predominantly at this stage just again describing 70% of the industry. We are predominantly in an area and in a landscape that has dune swale formations. So we have sand dunes running east-west, and then they graduate into swales, which are also running east-west, and then back up into sand dunes, east-west, and back down into dunes. So predominantly our orchards are planted north-south, so they go across the grain and across the elevation, and all those things, they're mixed together, and we have issues. Broadly speaking, some of the challenges in our industry are around water management, which I'll touch on a little bit later. All right, so that just gives you a bit of a picture of our soil types. And as I said, about 25%, and what the more recent and expanding part of the industry, so over in the Griffith region, is in these sort of floodplain alluvial type soils, which don't have these lighter textured issues. They have other challenges, but they're the two sort of broad soil systems that we're dealing with. Rainfall, very clear, very quite different rainfall. If you just look at the high level, I've got rainfall there for both Fresno and Mildura. That's in millimetres. What I've done throughout the presentation is done my best to try and present some of the data in imperial, but there is some hangover here into metric. So if you look at the top line there, there is very little difference in the rainfall. So at the first instance and first glance, you would suspect that California and Australia are quite similar. However, once you dig into the detail, which is the chart below, you'll see that the Californian rainfall pattern is more Mediterranean type, which is winter dominated. So again, for those Australian people present on this, I've normalized the rainfall, so it is like for like. So January for this chart here is July in our instance. So I've normalized it to make it like for like. So you can see there that's very Mediterranean based, California's rainfall, whereas we are pretty much in broad sense, an inch a month is our average annual rainfall in most of the key growing regions within Australia. So what does that mean? Well, it means that we've got summer rainfall. So we've got potentially 50 mils of rain around harvest in most seasons, and sometimes more, particularly recently. We have issues with rainfall around hull split, and obviously our friend nonpareil, which is very susceptible to hull rot, is very prone to infection and strikes within Australian orchards. So that little window there, which is again normalised for the Northern Hemisphere, is a real challenge for the Australian industry, and quite a different scenario for the California industry. Obviously, there's some exceptions to that on varying seasons. Sometimes we have drier years, as does California have wetter years. But on average, that little window there is a key difference between the growing areas. And if you look even further into the details, so this is just Mildura. I didn't analyze Fresno or other data within California, but this is the Mildura data. If you look at the mean versus the median for those statisticians, you'll see that the mean, for the most part, is greater than the median data. So illustrating that Australia can even have even wetter summers in some seasons. So what happens there is the tropical moisture. I'm not sure if it's similar to the Pineapple Express that you hear about and read about within your country, but we have tropical conditions that come in from the northeast and the northwest, and that sort of unsettled weather pattern, those unsettled weather patterns come down into our growing regions and can provide and be the source and origins of that summer rainfall. In terms of Australian production trends, I'll just sort of quickly explain and give some context of how we've evolved over time. You can see that for the most part, the Australian industry has typically occurred, and certainly the ideation of an almond orchard and various projects, capital projects, have often occurred in average or drier periods. As a consequence of that and various other reasons, obviously financial reasons and all sorts of issues as well and considerations, is that no one likes to see soil left unplanted. So everyone has tried to maximise their planted area for all obvious reasons. However, due to that previous first slide where we saw that variation in elevation and soil types and so forth, is that you can see quite a bit of variability in soil type that exists within our landscape. And the combination, you've got those competing tensions between variable landscapes and soil types and financial and other valid reasons to try and maximise your planted area. And sometimes you'll have instances where there's not enough consideration of soil types with what should be planted, and even if it should be planted, how should it be watered, and how should that irrigation design be set up. So on the picture there on the right is just a bit of an example. That area was planted. It has subsequently, those trees have subsequently failed through either, well, essentially anoxic, asphyxic conditions, and those trees have perished through one of a number of wet periods that the industry has encountered between 2010 and more recently. So as a consequence as well is that, or not as a consequence, but one of the features of some of the expansion here in Australia is not enough consideration has been given to surface or subsurface drainage. So we result in these wet areas, and trees eventually perish in bits and pieces throughout the orchard, throughout people's orchards. And I'll come back to what that means later. Other production trends that we're seeing, certainly the increase in hybrid rootstock, and that's largely a consequence of we are moving into more marginal soil types. And for us, the definition of marginal soil types are shallower topsoils with increasing alkalinity. In those sandier textured soils in the floodplain areas, there's other definitions for marginal soil types, but certainly we have started to venture into the hybrid rootstocks. But you know, for a long time, basically until probably 2014 thereabouts, give or take a year or so, depending on where you were in the journey. But up until that period there, the Australian industry was predominantly planted on Nemagard rootstock. And the other reason we're chasing the hybrid rootstock is to try and really chase vigor, and hopefully that's a proxy for water and fertilizer efficiency in these difficult economic conditions around cost of production. We are seeing an increased density within the orchard, tree density within the orchard. Like California as well, we typically started around 100 trees to the acre, and we are seeing most plantations planted these days of around 130 or more trees an acre. Trying to obviously reduce the duration to maturity, and trying to increase yield efficiency and uniformity by sharing the load amongst many more trees. We're trying to, you know, for the most part, pruning has dropped away, really for two reasons: labour costs, as well as trying to reduce the time to mature yields. So for us here in Australia, in US dollars per hour, we're about $27 to manually to send someone out there with a saw or some description to go out there and with a pair of snips and start manually pruning trees. So for us, that's expensive, and we as a consequence, trees these days have gone from 3 to 4 main scaffold limbs to 6 and often quite a number of more than 6. So we're certainly seeing a more congested canopy in our more recent orchards. We've had a slow uptake of self-fertile varieties, largely because of a higher incidence of bacterial spot. This has actually happened with Fritz. So if you come to Australia, you won't see a lot of Fritz planted either for the same reason. So bacterial spot is a bit of an issue here, maybe attributed to the summer rainfall that we experience. So as a consequence, for a number of reasons, we still see nonpareil the major part of Australian orchards, certainly around the 50% of plantings, and even more the more recent plantings. There's still a slow uptake with self-fertile varieties. As a broad generalisation around bacterial spot, apologies to those that think they don't have susceptibility for self-fertile trees, but several have in our experience. We're certainly seeing a more intensification of inputs. So we touched on it before, but we're drip irrigated orchards. So at least 95% of our plantings here in Australia are under drip irrigation. And we are seeing regular, again, attributed to our sandier, lighter textured soils for the most part, but also more broadly because of an objective to intensify our water and fertilizer and try and improve our yield performance. So we are seeing 270 pounds to 290 pounds an acre of nitrogen annually. We're seeing foliar applications of nitrogen and other nutrients as well, mind you, and daily applications of water from March through to October in Northern Hemisphere times. And we say daily fertigation, but typically it's 3 to 5 days a week we'll be applying nitrogen. So it's lower dose, but more consistently through the orchards. And you can see on the right there, you do create these really large sort of intensive fibrous root systems, which we think have got some really good advantages when it comes to yield and performance. However, there may be some knock-on negative effects around high humidity within the orchard and underneath the canopy, disease risk, and potentially pest management issues as well. New fungal chemistry is expensive in Australia. So we have an agency system where patented chemistry is sold under high fixed pricing. So in Australia, that can be north of $150 a litre. So it, access to some of the newer chemistry, which is at least got suppression characteristics, if not preventative type characteristics against things like coal rot. It's difficult to try and justify, particularly at the moment with the current cost of production, and it becomes difficult accessing some of that new chemistry under those arrangements here in Australia. And back to labour again, winter sanitation is expensive again because of the cost of labour. So polling mummies remains an unaffordable activity within the Australian industry, and for the most part, our sanitation is mechanised with additional shaking and additional harvest passes. So Carpophilus beetle was found, and this is why I draw your memory back to that first slide. So we're in the southern Murray-Darling Basin. So we're in the sort of southeastern corner of the country. We're basically the 3 states joined, so the borders of South Australia, Victoria, and New South Wales. So just in here is where the majority of the industry is, and then more recently there's an expansion up here in New South Wales around Griffith. So we had a wet period. So in Australia, broad, very rough, broad terms, we have a 10-year weather cycle where 1 to 2 years have been historically quite wet. 1 to 2 years within a 10-year cycle have been historically very dry, and then a range of sort of average to, above or below average in between. So the Carpophilus beetle in the Australian industry was really started to, was identified following these 2 wet years. So in terms of identification and sampling, I'll talk on our experience. So we're a vertically integrated participant within the industry. So we grow, process, handle, sort, pack, and trade almonds. So we operate across the full supply chain. So whether, I guess, you're a vertically integrated grower or not, or you're a grower receiving information back from your handler, we quickly worked out once we started to see more than one insect. So as we've already discussed, historically, we've always had an issue with carib moth. But the issue, I guess, when you start to see some of the information that gets delivered back from your handler is it's just insect damage. So by the time that the kernel finds its way into the sizing bins and the sorting and packing sheds and so forth, all you're seeing is the pinholes or the various tracking that might be occurring on the surface of the kernel. And it just comes back to the grower's insect damage. However, once we worked out that there was more than one insect involved in some of that damage, we quickly went back earlier in the system and certainly would encourage growers to try and make sure that the identification and sampling is done as early as possible in that production system. So obviously you've got your pest and disease scouts and advisors and so forth. So that's all fairly standard practice, but just to really emphasize when you are dealing with 2 different insects with 2 very similar sort of symptoms, once all the identifiers are removed, so the frass, the webbing, whether it's granulated sort of frass or fine powder like a beetle, so that they are quite different, symptoms early in the system that are easily visible. But so, again, at the end, it just becomes insect damage. So certainly encourage everyone to try and sample as early in the system as possible, and try and work out what insect you have, because there's certainly different management options available to you, other than obviously sanitation being a unifying approach. The other thing I guess we found as well is that as we've followed the damage through the system, we've started to paint a picture and put some numbers towards the damage and the financial impact it's having. We are certainly seeing that the damage is far worse in the orchard than what the handlers are reporting, because some of those compromised nuts and kernels that have been half chewed or etc., they do not see the light of day. So particularly Carpophilus is quite aggressive in its feeding. So some of those damaged kernel are being aspirated off in the hullers and shellers, and you're not seeing all the damage when you get your reports from your handlers. So another reason to try and go back earlier in the system to get a better idea of the damage that's being occurred and the financial impact. Certainly encourage everyone to get granular in your data. The Y and X-axis have been purposely removed for this presentation because of sort of being a bit sensitive. But the X-axis is a range of different farms within our group, and the Y-axis is the varying levels of damage that we're seeing attributed to Carpophilus beetle from our on-farm sampling. So like David Madge was indicating, we sample after shaking, so try and get the best representation of the fruit. So we don't sort of do too much of our sampling other than through the year, but once we get our, when we try and do our proper, detailed insect damage assessment, we do it post-shaking once the fruit's on the orchard floor and we've got a decent sample size and representation. So certainly encourage you to get granular in your data. Not all of our orchards have got problems. Some have. I guess the thing I'd like to highlight here is the hotspots. So you can see that the top whiskers on these box and whisker plots, you can see that the means might be okay. Again, always coming back to granularity in your data. The means might look okay, but we are finding that there are hotspots within orchards. So again, I'll draw you back and draw the thread back to some of the slides I've already shown about the variability in our landscape, the topography, the wet area and a lack of drainage considerations, drip irrigation and intensification of watering. For us, our hotspots are all those things there. So you see nuts on the ground, free water on the soil surface, weed management getting out of control, shallow or heavier soil, hibernating or retaining water within the soil, etc. Really encourages, in our experience, high levels of Carpophilus beetle. So just be aware that means and average data might look okay, but there are going to be hotspots. You need to go find them. Some of the discussion earlier on was cohabiting of the two different insects. For us, we've got carrot moth. We don't have navel orange worm, but it's quite similar, and beetle. So these are a range of our farms, and the variation in contribution to insect damage. So on the y-axis is 100%. So that's obviously not 100% damage, but of the damage found. So for example, column 1, 80% of the damage found was attributed to carrot moth. In the on-farm sampling post-shaking, pre-pickup, 80% of it was contributed to carrot moth, and nearly 20% of it was contributed to nuts where both insects were found in those kernel samples. So you can see a whole range of and spread of varying scenarios. So again, I guess the message for me is to again get granular in your data and understanding which insects on which farms, in which areas of which farms are causing you the damage, because it's not always going to be Carpophilus. It's certainly, we've been on a bit of an emotional roller coaster here in Australia. At various stages, Carpophilus is completely stolen the oxygen within the conversation around insect management. In other instances, it's dropped away wrongly. So it's just trying to have a balanced, objective discussion of what what insect problem we've got. Is it beetle? Is it moth? Is it a combination? Is it something else even? So management implications. So it's a busy slide here, but basically challenge number 1 is summer rain for us. So we have 1/2 inch to 1 inch of rain at non-pareil hull split, and in harvest we can see up to 50 mils. And in the last couple of seasons we've seen rainfall in 100, 200 mils through harvest. So in those conditions, certainly there's implications. So high incidence of hull rot, we get delayed and certainly protracted harvest. So obviously we get interruptions because we will have some rain and then we can't harvest, and then we've got to wait for things to dry out, and then we start again. So all those things certainly attribute a higher level of Carpophilus. Carpophilus, like most insects, they are attracted to the early splits. I guess I don't necessarily say this on this slide, but certainly we are finding Carpophilus on end trees, outside rows, etc. They are attracted to early split fruit, and they are very much attracted to the whole rot fungus. So whether it's the volatiles and or the being a food source, but they are certainly attracted to whole rot. And certainly with the protracted and delayed harvest, clearly that leads to more time for infestation and damage. So I guess what we've been thinking about in terms of management applications, certainly deficit irrigation, low nitrogen, these are all common things around whole rot management, as everyone's well aware. Whole split fungicide applications, but as I said, there's some complications for us around affordability in that space in Australia. The big thing, I guess, that we're looking at is the date of shaking. So for us, that we think that's the greatest opportunity at the moment to have the best impact. And our data tells us we need to have nonpareil shaken by the 1st of March, so it'll be the 1st of September in the Northern Hemisphere. We need to have all nonpareil off the tree by that date. You have nonpareil on the tree after that date, and you will see your damage from Carpophilus beetle exponentially increase. So we're trying to start to shake as practically as possible. That obviously has the complications of all the fruit not necessarily being ready. And again, I draw you back to some of those earlier slides I presented around variability within the landscape. We have variability within fruit maturity, not just within the canopy, but certainly within rows, across irrigation valves. So the date to shake, like most orchards, I don't think that's too different in California or Australia, is difficult to try and pick. And at some stage you have to make a call. And for us, we are certainly erring on the earlier side of things. However, in doing so, you're going to have green fruit left behind, as we all know. And as Dave was indicating, if that fruit doesn't come off, you're going to have that fruit. You might have got away with it this year, but that fruit is now going to provide you a problem next year. So we are typically doing 2 passes for nonpareil. So we will shake, go in as early as possible, and then a second pass for nonpareil harvest as well. We'll do winter sanitation. So subject to our ability to do that second pass on, which is basically linked to weather, we will do winter shaking. As I said, we can't afford polling, so we'll shake, and then we will winter pole. We will shake, and then we will do spring and summer pre-harvest sanitation. I'll talk about that in the next couple of slides, what we do there. Again, no surprises, but it's, your flower mowing and all those types of things that the Californian industry does as well. Supply chain sanitation. So it's not just what happens on the orchard, but we'll talk about that quickly as well. So as I said, wet area management is a key thing for us. So whether it be surface waters, water tables, weed management, uneven fruit maturity, higher mummy loads are the consequence, leading to higher levels of fruit on tree, which become fruit on ground. And fruit on ground next to drip irrigation that sees water delivered pretty much daily for the majority of the season leads to higher moisture levels and sustained levels of Carpophilus populations. So management implications, we're obviously regularly looking at surface and subsurface drainage options to try and improve water management. We, in some instances, are redesigning irrigation valve boundaries. One irrigation valve can become 2 and possibly even 3 irrigation valves, but certainly 2. We've done that on 1 or 2 properties. It's not a common thing, but we have done it. Soil water management and irrigation scheduling is obviously important around deficit irrigation as well. We control extra sanitation, which I've touched on. So for drip irrigation problem.
Sorry, Ben, if you could wrap up within about 5 minutes or so, that'd be helpful. I know you have a few slides left, but
no problem. Thanks, Katie. So the other thing with drip irrigation is, in our dune swale formations, is the drainage of the water out of the submains and mainlines into those low-lying areas. So water management out of the pipe work is a key thing. Shell seal, David touched on, so I won't go into that too much more. We did some trials around insecticides. Really, the key thing here, as you can see on the right-hand side, is that we're not seeing a lot of benefit at this stage that justifies insecticides. And again, most of the chemistry that's on this screen here is quite expensive for us here in Australia. So the combination of the two is making uptake of insecticides slow in many instances. We've tried some entomopathogenic trials as well in conjunction with David around and seeing if we can see a improvement in damage there. You can see the 3 controls are not necessarily, or the treatments are not necessarily seeing a benefit over, that untreated. So early days yet, but certainly a space that we're quite keen to monitor. So just lastly on orchard sanitation. So for us, we're looking at the whole, obviously, supply chain. It's not just obviously at the time of shaking. So whether it be equipment, making sure everything's ready, whether it be shaking. Obviously, as you can see in the picture there, you go in early, you're going to have fruit left behind. You need to go back and get that fruit as a second pass. Obviously, we need comprehensive nut recovery from sweeping and conditioning and harvesting. We'll do a second harvest. And then sometimes we'll do a further winter reshake. And then we'll do some orchard floor sanitation. Because of that fruit, as you can see in the picture there, that sits around the drip line and can become an issue, well, certainly is an issue with hotspots and Carpophilus infestation. Quick trial we just did it on our own farms. These are not highly scientific, but you can see the benefit that everyone's well aware of between orchard sanitation. So this was just nonpareil only that was reshaken versus all varieties reshaken. So you can see that we certainly have seen a positive impact on orchard sanitation. So the key thing we're probably looking at is the effectiveness of the orchard sanitation. So we've got a product here called the Almondator. So Select Harvest and a group have worked together to develop this technology. It sits on the back of a conditioner. So product is picked up, cleaned, put through the impact rotor and flail mower, and spits out very fine particles. So we have seen good success with this technology because we've just find that the flail mowers on our sandier soils are not working quite as well. There's some pictures of before and after, a fair bit of leaf and trash on the left. So next steps. So at the moment, we're operating in somewhat of an even environment. We're going to try and get that a little bit more even for not just insect management, but certainly some benefits around yield as well. Shakers, the newer shakers are certainly getting more fruit off. We'd like to see some more investment in them as time goes on. And David's talked about the monitoring and the attract and kill. Thanks, Katie.
Yeah, thanks, Ben. We can do about 3 minutes of questions. I'm gonna launch the 3rd DPR quiz. So if you need those DPR CEUs, make sure you respond to this quiz. Yeah, again, I'll give it about 3 minutes. Let's see the questions. Thanks for that, Ben. That was really thorough and super informative.
Can I just ask a quick thing, Ben, to clarify, the entomopathogenic fungi trial you did was just with commercially available products?
That's right, David. Yes.
Yep.
Yep.
Let's see. I'm gonna do this quick insecticide question that Ben Wiblin answered, but I thought the answer was really good. Why is clothianidin typically avoided due to being a neonicotinoid, MRLs, or might flare up? And you said all of the above, so that's a good thorough answer. Okay, it looks as though it may be possible to differentiate NOW frass from the beetle, but the beetle frass appears very similar to ant frass. Any thoughts there? Do Australian growers deal with ants?
Ooh, I replied to that one.
Yes.
But yeah, basically, or actually, Ben, you and ant control.
Yes, we do deal with ants. We probably don't give it enough priority. It's probably fair to say. that there is opportunity for us to do more in the ant space. But yes, the frass is probably quite similar, fine, flour powder like frass. But certainly, obviously, at the time of collecting the nuts off the orchard floor for our sampling process, that's part of the identification, I guess, if ants are present or not, and if they're not. But it's rare for us to see damage and not see the insect in the orchard. So once we're sampling off the orchard floor, it's rare that they have exited the fruit. That they are normally there. Yeah, so they're not difficult to miss.
Yeah, a California answer to that question is if you had a pile of frass from southern fire ant and a pile of frass from Carpophilus with nothing else to look at, you're probably not going to be able to tell the difference. So that's where the other things that they've mentioned come in. Are there tunnels? Are there not? Of course, the ants only eat the white part. They leave the pedicel behind. And so you'll see kind of that empty skin left over, whereas the Carpophilus tends to kind of bore through back and forth. So you got to look at other things. But the other reason that's important to say is identification in the field is very different than identification in a USDA hull sample. Because, once the hull's off, the shell's off, the frass is gone, if that kernel wasn't obliterated and all you're looking at is what's left over, the frass isn't going to help you. So you have to look at those other symptoms to tell the two apart.
Okay, yeah, that all sounds good. Thank you all for answering all those questions. And yeah, thanks for paying attention to the Q&A box. To just keep us on schedule as best I can, Ben Wiblin, I'm gonna go ahead and let you go. I'm gonna end this quiz. But yeah, feel free to go ahead and share your presentation.
Thanks, Katie.
Yeah.
Have you got that all right?
Yeah, looks good.
Great, thank you. Yeah, good afternoon, everyone. I'm sure we're all sitting through a bit of webinar fatigue just at the moment, so do bear with me. I think my slides, probably not as many, but probably do provide a bit of contrast or context into, I guess, what our growers are dealing with on the ground, but also from a financial perspective as well. So I work for a company called Almondco Australia. So we're a packer of almonds. We're a co-op established in 1944. We currently represent about 85% of the whole Australian almond growers, processing around 90 million pounds per annum currently. And that's sort of on a pretty steep trajectory up to 110 to 120 million pounds by the end of the decade with, as you can see, they're a pretty strong export focus. We do offer our growers both hulling and shelling services along with secondary processing and packaging of brown kernel lines and blanch and value add lines. But I think the most important part of this slide is probably just to demonstrate to you guys that we do cover a lot of the industry. We do draw kernel from all growing regions across the country, and we are represented from both family farms all the way up to some large corporate entities. So from my time across the last couple of years, driving up and down the valley during harvest, the Central Valley, I certainly got a pretty quick idea that you guys do enjoy the data. You want to see who owns the data, where the trends are, what's plotted against what. So I didn't really want to muck around this morning, or sorry, this afternoon. I want to get straight into it. So you can see the Almondco average over a 10-year period is at 0.77% insect damage across all varieties. I guess a caveat before I start, and Ben Brown alluded to it previously in his presentation, is that like the US grading standards, the Almondco grading standards do not take into account Carpophilus Beetle specifically. Insect damage is pooled data, so it encompasses all insect damage. And yeah, it isn't differentiated by insect species effectively. So What we do know, though, is that Carpophilus beetle is absolutely one of our primary pests, contributing anything up to 50 to 60% damage each year. So of that 0.77% average, you can imagine that Carpophilus beetle is contributing pretty significantly to that. Probably the elephant in the room, though, if you look at the non-pareil column here, that's averaging 1.23% damage over the 10-year period. So a pretty stark contrast in comparison to the average of all our other varieties sitting at 0.32%. So what we do know, and I think this question popped up a few times in the question box, is it is a non-pareil problem, but from a big picture point of view, across all varieties at 0.77%, I believe it's a pretty well managed problem at the moment. Of course, at 0.77%, we always want to do better. So if we try and jump into the data a little bit closer and unpack that, we can see here, I know it is late in the day, everyone, so graphs are probably getting a bit beyond everyone, but the blue bars here are the annual rainfall since 2016 for one of our key growing regions, moving through to 2023. The red line here is average rainfall for that period, and then the green line is the relative insect damage that was incurred in that year, respectively. So you can see there's two pretty clear spikes, one here at 2018 at 2.5%, and then another one in 2023 at 1.7%, respectively. So what also accompanies those spikes is two pretty clear spikes in rainfall or annual rainfall. So you can see in 2016 about 13 inches, and then 2022 got absolutely soaked and got just over 18 inches of annual rainfall. But like Ben Brown alluded to, unfortunately, our environment doesn't permit that rainfall only falls in the winter months, and we do get a portion of that during harvest. And then what that leads to and what that means is effectively we don't harvest our orchards on time. The harvest gets delayed. We get poor timing of shapes, and then unfortunately we get a higher mummy loading, and now due to orchard accessibility, the sanitation programs just don't get done to a satisfactory level, and then consequently in the following year or latter year we do see these spikes in insect damage. So although we never like to see it on the back of high rainfall years, we do expect to see insect damage lift. So this is, I'm sure if we've got some growers online, I think this is sort of a bit of an area where you guys might be interested in, and that's how much is it actually costing Australian growers at the moment. So the top table here probably looks pretty similar to what you're used to, but these are the Almondco serious defect grading parameters for insect damage. So you can see across the top, we've got the tolerance and then the grading charge below. So we do allocate a bonus if you come in equal to or less than 0.5% insect damage per annum. Of course, there are a few other grading parameters stacked into this. It's not just insect damage, but you can see as you move through the damage levels from 0.5% up to 5%, you do start to incur from 2 cents to 10 cents per pound penalty. So that does start to kick in. And of course, similar to you guys, all insect damage product is classed as inedible. So it's not a saleable product. So effectively, that's a direct cost to the grower for delivering that product. They will not get paid for the weight of that product, of that damaged product. So the blue bars down here represent the annual dollar cost to our grower pool since 2013, so a 10-year period. We've got the average cost of insect damage. Keep in mind, that's not just Carpophilus beetle, that's insect damage as a whole, sitting at about $1.3 million per annum. So again, it's still $1.3 million, but at the moment, I believe it's relatively well controlled. Of course, we do have these spikes in 2023, which you can see here. So I think something that's not portrayed in this graph are the indirect costs or the on-farm costs that are being incurred of managing the beetle. So if you're at 1% insect damage per annum, you're receiving a 5% per pound penalty on your crop, but you're also investing in sanitation programs, fumigation, spraying. These costs are actually a fair bit higher. So I think that's certainly one thing to keep in mind. Probably the other thing just to keep in mind is that at $1.3 million, that's still less than 1% of our total grower revenue pool. So still relatively under control, I believe. So that was the data, and probably I just portrayed probably a pretty clear message to you guys that perhaps it's not as big an issue as what you might think. It's certainly a very well managed issue for us at the moment, but you probably just need to put that into a bit of context. Although our growing practices are pretty similar, we grow similar varieties. The reality is the environment that we're doing so is still significantly different to yours. So Australia alone, we're producing almonds across 65,000 square miles. The Kern County alone surpassing our total acreage in just 8,000 square miles. So you can see there's a pretty stark contrast here in area. There's a high, monoculture of almonds throughout the valley, a highly densified planting throughout California generally. And unfortunately, this could just be the Achilles heel, I guess, that we're not dealing with. And it's a problem or a challenge that you guys will deal with that we won't. I think just building on that as well, I read the monthly reports that come out about crop progression out of the US. And we always hear about, the abandoned orchards, orchards being removed across the I-5 and that sort of thing. And that is another challenge that we don't have. So those orchards are being left abandoned or awaiting removal and simply just harboring pests. That isn't a challenge that we deal with, but it will absolutely be a challenge for you guys to deal with. And it's probably something that we can't wrap our or we can't probably apply any numbers to just at the moment. Again, although the numbers do suggest that perhaps the beetle isn't, incurring us a heap of damage from a higher view point of view, absolutely, like Ben alluded to, there is absolutely hotspots. We've got some orchards, reading up to 10% insect damage per annum. But on the big scheme of things, all growers combined, our insect damage is still sitting under 1% overall. But that is on the back of our growers tightly managing that. And I do apologise for this picture. I know it's pretty ordinary. Take it off there. But I did just want to show you this tractor with the V-sweep attached to the front, followed by a mulcher, a flower mulcher, followed by a blower. So that's a pretty efficient one-pass sanitation program. But I guess that is also coupled with the grower knows where their mummies are. So they do perform mummy nut mapping. They know the orchard topography. They will do reshapes when required. They'll go in green and harvest a bit earlier if they can. And of course, they're trying to manage their nutrient loading coming into harvest, and they will use agrochemicals as required, but they will also avoid having to spray if they don't have to. Sanitation, I'm not going to go into sanitation. You guys taught us how to clean up orchards. You've been doing it for a lot longer than we have. I've listened to plenty of presentations from both David Haviland and David Madge about the importance of this. Effectively, it will just simply underpin all integrated pest management programs that you invest in. It's important just to simply do the fundamentals well. It will certainly go a long way into managing the pest. So this graph here plots whether or not our growers apply insecticides or pesticides, not just at harvest, not just for Carpophilus beetle, but at all. So interestingly, you can see here that only 19% of our growers are actually spraying any form of insecticide or pesticide on their orchard. So generally, most of them are purely relying on sanitation programs. And I guess just for this data is pretty rigorous. We do audit 100% of the spray diaries for growers that send in their almonds to us. So this is pretty true. I guess one thing that we have seen in more recent times is a movement to in-field fumigation or field weight fumigation, which is probably being pushed more and more from our end. The problem with that, and like David alluded to, the efficacy of using phosphine at the moment just doesn't do a sufficient job on killing the beetle. Of course, if it does come in contact, it will do the job, but trying to retain the concentration for a long enough period just isn't possible at the moment. So we do recommend that growers use sulfuryl fluoride and bring in a trained a trained expert effectively to do that fumigation service. These are the 3 chemicals that are currently being used on farm, or they're registered for use on Carpophilus beetle. Again, typically clothianidin is avoided just due to the harshness of the product. Actual efficacy of the other 2 products or 3 products generally, I guess the jury's out. But if you're getting 1% or 2% effectiveness, and I think it's a bit of a cumulative effort trying to drag that insect population down. We'll recommend pulling out all stops if damage levels get too high. So I certainly don't want to make a mockery of a pretty serious topic this afternoon, but we often do refer to the beetle as a storage piranha. I guess the key takeaway from this slide is it's simply just not a field pest. It's absolutely a storage pest. So we spend a lot of time and do a lot of work eliminating residual almonds, making sure that we don't have areas around the storage facility that can harbour pests. We'll always isolate hot bins or infested areas, and we'll continue to monitor and fog the facility as required. Right, so I guess these are just a few steps that we have in place to keep insect damage as low as it can be. Field weight fumigation, it's a little bit different, our intake program compared to yours. So in Australia, our growers have to store their almonds on the farm awaiting for hulling and shelling. So the model is slightly different. In the US, you guys all just send the kernel straight to the huller and sheller and they fumigate it and manage it for you. That's not the case in Australia. So if you do have insect populations within the orchard, they will carry over and survive in your stockpiles. So it is important that the field weight does get fumigated as quickly as possible if you do have activity in there. So once received and hulled and shelled, we will fumigate 100% of the crop before it enters our secondary processing or packaging facility. We use fumigation chambers using sulfuryl fluoride. And then following that, it moves into our secondary processing facility, goes through a series of hand and optical sorting, and we will use a pre-dispatch fumigation if required. Right, so that pretty well summarises my sort of pretty high-level overview. I guess just to finish, although I probably deflated the situation a little bit, can't take away or can't stress highly enough that this is a highly destructive pest when not managed sufficiently. It'll absolutely attack both field weight or crop in the field and in storage. Without a rigorous sanitation program, the beetle will absolutely get away, and we see that time and time again in our orchards. But finally, hopefully this makes you sleep a little bit better, that it is manageable. We are currently managing it, but unfortunately, yeah, doesn't come without a challenge. Thank you very much, Katie, and everyone else, and certainly open to any questions.
Cool. Thanks, Ben. That was great. So for the attendees and everyone, really, that was the last presentation that we have to share today. So before you hop off, I have one more quiz to share for DPR, but more importantly, this can just be an open discussion. The webinar was initially set to end in about 15 minutes, but we'll just see how long questions go. And I think it's helpful to just kind of pull everything together here, have a good discussion. Yeah, definitely want to ask some of these questions that haven't been answered yet. So the first one, oh, actually, I need to share the poll first. Let me share the poll and then we can, okay, so the final quiz is up. So would it be possible to develop a sprayable repellent, or does maybe mating disruption exist for this pest yet?
Oh, quick word on that one. So Carpophilus are known to produce an aggregation pheromone rather than just a straight sex pheromone. So the aggregation pheromone attracts both species. And the idea, like I said earlier on, is to basically say, I want a good food source, come on in. Mating disruption is a possibility. It wouldn't actually be mating disruption, it would just be pheromone communication disruption. I don't think anyone has tried that. I don't know of anyone who's considering it. So I couldn't actually comment on the likely feasibility of that.
But David, that would be more feasible for attracting kill than for mating disruption, correct?
Yeah, I think so. And that's why we're going down that track. And while I've got the stage, I'll just make one comment. Something that we haven't actually touched on is that in Australia, this is a really damaging pest. It may be that in California, because of different chemicals that are being used, the different chemistry, different application rates, different hygiene levels, soil types, maybe an environment, it may be that you have the beetle, but it just operates at low levels and doesn't cause you grief that it has caused us. We don't know yet. Until you can monitor it well and monitor harvest damage levels well, we won't actually know what it's doing over here. We just know that you've got it. So it's a kind of a wait and see to see whether it turns into a major pest or just a background thing that is kind of under general management, because you're doing so much for navel orangeworm already.
All right.
Probably the other element I'd talk to there, David. I know we kind of push you a little bit about it, but yeah, it all comes down to what David's working on at the moment with efficiencies in terms of how many traps you need per hectare, what's the cost of this regime. They are looking at different ways of distributing it and how they can, I suppose, control the pest, whether it be through a bait system that maybe or something else. That's pretty well where we're at this minute.
So Katie, can I jump in and ask Ben a question?
Yeah, go for it.
Sure. Yeah. So Ben, sorry, the final Ben, got two Bens here. So you talked about how Australia's got about the same acreage as Kern County, but you're spread out, we're condensed. The other thing that hasn't really been mentioned on this call is pistachios. Obviously that's not you're obviously almond people. We've also got pistachios in the mix. And we've got a case where we've got the beetles in some places, but not others. And so I think you saw a trend in the questions. There's a lot of people asking questions about, does it move from one orchard to the next? How far does it fly? How quickly is it going to move from one county to the next? Can you just kind of summarize your thoughts on this, how fast it moves, both regionally and from one orchard to the next? How big of a risk is one orchard to its neighbor?
Yeah, no dramas. I think in terms of speed, perhaps David or the other Ben might be a better person to talk to about it. But absolutely, if you've got a harbouring orchard next door, they certainly don't stick within the refines of their boundary. They'll be happy to jump the fence and visit the neighbours for sure. So I think it's absolutely a risk. I think the other risk for you guys, you know, like we move a lot of product around the place. You guys have got, you know, over 100 hullers and shellers. You've got product moving all up and down the 99, the I-5, left and right. So irrespective if you've got a neighbouring orchard, if you're near a huller, if you've got product moving around the place, you know, you're just going to naturally be moving the beetle from A to B. So I think the risk is greater for you guys just due to the, you know, the densified sort of nature of the plantings of both pistachios and almonds. And now, you know, they're not only neighbouring, you've got the hullers and shellers now. That sort of dynamic adds to it as well. So I don't know if David or Ben wants to add around the speed, do you think, or the nature that they fly. I know that sort of popped up a few times as well.
Okay, so we haven't done any measurement of their flight capabilities. Carpophilus can fly miles and kilometers. Our observations in the orchard are that, yeah, when they're being disturbed, they'll spread. So I have heard anecdotal notes of one orchard harvesting and shaking and beetles coming en masse into their neighbouring property. But we don't have any evidence of like mass migration or anything. They just shift around when they've been disturbed like that. I think we probably do a fairly good job of shifting them around ourselves, really. So like you're saying, with the just movement of crop and equipment and so on, that as far as speed of, or not spread, but growth of our issue here, that we went from, in 1 year, I think I saw a nut that had damage in it, to the next year 7% damage, to the next year the industry called an emergency meeting because they were getting their own measurements were showing 30% damage in some blocks, and not just one farm, it was like multiple farms in that region. So it went from nothing to big bang very, very quickly. And it'd just be interesting to see. We don't know what's going to happen in the Californian industry because we know that I think it's been here for at least a couple of years. So I imagine if it's going to be shuffled around, it's been shuffled around for a couple of years. Whether now that we know that it's here, whether you're monitoring at processing plants or at harvest in orchards shows you that you've got 0.1% damage, who cares, or 15% damage in some areas. We won't know that till you get the data.
Thanks.
And David, probably just to add to that, it's a bit of a duplication, but for us, I guess we've got 15 orchards, so certainly moving machinery around for us is a conscious thing. So we do treat the insect, even though it's endemic, as a bit of a biosecurity issue. So I think there's a fair bit of contract harvesting done in California, but we don't, but we move our own gear around. So I would certainly, encourage growers to look at managing it as a biosecurity risk and trying to avoid shifting it around, because certainly that's where we have found it. It can be shifted around quite easily.
In terms of a industry level, just to final out, I guess, it's probably not really relevant, but what we've seen in Australia is with it starting in Sunraysia in 2011, it has spread to other regions, probably the most like South Australia in our Riverland region, probably hasn't been exposed to it as much over that time period, but I think it's starting to come there now. So that's like 200-300 miles away from the Sunraysia region. So if you want time zones, well, obviously distance is the key one there. That's probably affected that timeline. But yeah, you'd expect it to get you some point.
And just on that, we know there are orchards in the Riverland in South Australia where we have detected the beetle 6 years ago, and it hasn't actually taken over. So that's something else that needs more looking at is why it fires up in some orchards. And that relates to a query I wrote down to do with Ben Wiblin's talk. The difference between Ben Brown, large corporate grower, and Ben Wiblin, who Almonco deals with a lot of smaller to mid-sized growers. A lot of those growers will tend to be smaller properties and potentially a little bit more isolated. And what we've found when we're looking at carrot moth early on and doing searches for infestation in mummy nuts and that sort of stuff, any growers who's kind of a bit smaller or slightly more isolated, depending on their environment or nearer the river, they tend to get cleaned out. Their mummy nut crop gets cleaned out by birds very effectively. So you can be lucky. You could be in a potential beetle infested region or district. But if your crop residue is cleaned out naturally by birds going through in winter, you may not actually develop populations that end up being damaging. And there's a big difference between that and the areas where the big corporate farms are, where it's like 10,000 hectares of almonds in one lump. Very big difference and very big implications there for what levels of damage they end up with.
Oh, Jalindra, you had
Yeah.
your hand.
So I just wanted to provide the comment, the same thing that more or less similar that David mentioned about pistachio, and certainly if you have seen anything on walnuts, we found the Carpophilus truncatus in walnuts, but that was the sample collected after the harvest. So it was not active on the tree. Those are the sample collected from the ground after the harvest. So it at least it confirmed that they are in the walnut orchard. But at the same time, when we looked at the sample, some of the older variety with the softer cell, or has a little bit more poor seal, those are one had more damage, and then Chandler basically did not have damage from the beetle. And I've heard that in Australia and also Italy and Argentina, don't they find these beetles infesting on walnuts? Can you provide a little bit of perspective on that side of things? Because there are also walnut industry members also present in this conference, and would be interest to all of us.
Yeah. From the Australian perspective, we've had our hands more than full, so we have not gone out to the walnut industry to see what they've got to do sampling. That's something that needs to be done to see if it's there or not. And certainly in Italy and Argentina, they've shown very clearly that they do like walnuts when they get into them. I would assume that if we start sampling walnuts, we would possibly find them. We do have them in pistachios, but they're not considered at this stage to be a major pest. But obviously in California, that's where I think they were first noticed, was in the pistachio crop. So yeah, it's one of those things where if we start looking, I think we'll probably find.
In almond, certainly we're seeing the damage. The highest I've seen is around 20% from the Carpophilus beetle in some of the orchard. So that was last year, 23. So that means that, certain area is definitely, they cause a lot of damage. I think the dryness that we're thinking about, maybe it definitely varies by the region. Maybe the down south, maybe a little less problem, but especially when I'm at northern San Joaquin Valley, with the irrigation, all the cover crops and weeds and other things in there, and the ground environment a lot different than what we think, aerial environment. So, yeah, what I've seen so far, it seems to be pretty scary in many almond orchards.
Yeah. So is that damage level on a block basis or an average across a farm?
It's that particular orchard where we did the sample. So we have like 20 acres and then the 15 acres in different variety complex. The one that we had was nonpareil, Monterey, and Carmel mix. And nonpareil and Monterey had pretty high damage. The Monterey had over 20%. Nonpareil was about between 10 to 15%. And these are the sample collected just after the harvest. So not from the harvest sample, but after the harvest. So there might be a little bit over, estimated, but it's still, regardless, when you go right now, these orchard, you can see the beetle everywhere. We put the navel ongum, the trap with the bait, pistachio bait in it. And then we start catching these beetle like 2 weeks ago. 3, 5, 10 in these navel ongum traps. So they are certainly active there. So yeah, I think, but I agree that maybe depends on the year and other circumstances. It could be differences there, but certainly it's established and causing damage.
Yeah. So hopefully it doesn't go there, but it sounds like you're on a similar path that we were on.
Jalindra, that trap, so is that the ground pistachio meal, like a Peterson
Yeah,
trap you're talking about?
Basically Peterson trap, I
Right.
put it in there. That's where the adult beetle that's another interesting thing that we start catching them 2 weeks ago. Although I put the traps, in May. I think we put the traps in April, May, some point. So that shows there a little bit of movement. Also, 2 of the consultant in the area sent me the picture of the fresh knot with the beetle with the hole on the cell, like yesterday and today in 2 different areas in 2 different counties, Merced and Stanislaus County. And I was thinking that it's too early for the beetle to cause the infestation, but I was wrong on that assumption, at least, because clearly it shows that they can be active as long as hull splits.
They're there, they're flying, they're active. The second the hull splits, volatiles get released. It's a magnet, they go lay an egg. That's exactly what navel orange worm does.
Yeah.
It's exactly the same thing. It's all of those volatiles coming off the splitting hull that's attracting them. So interesting.
I want to make sure we address all these questions in the Q&A as well. So this one says biopesticides component lead for AgVic Australia here. In Australia, there's only one product currently registered that contains entomopathogenic fungi, which is Bavaria bassiana, that is formulated as a, quote, broad spectrum, unquote, biopesticide for insect control. There's also a product available that contains 3 different fungi species with unknown host specificity, but it is formulated as a soil conditioner and not registered specifically for insect control. So the EPF species formulation and delivery strategy need more research to target Carpophilus effectively, which is underway. I guess that's more of a comment, but if anyone has comments about that.
I'll just add to that. Yeah, Amy's the person looking at EPF within the IPM project, and she's doing a really good job there. But like I said earlier on, it's early days. Just to reiterate something I said earlier on, that when we've done bioassays of entomopathogenic fungi against the Carpophilus truncatus, the strains or the isolates of the fungi that we've got from almond orchards have given really high levels of mortality, like 90% larval mortality, much greater than the commercial products that you can buy. And it sort of makes sense that if you're getting a fungal strains from an environment where the beetle's living, maybe there's just more, I guess, more association between the fungi and the beetles rather than a product that's been developed from overseas to tackle different insect species entirely. But we've been kind of buoyed with the optimism with the levels of mortality we've got in the lab. We just have to see how that translates to being able to do something out in the field. The whole field application side of things, keeping the fungi active for long enough to infest beetles as they're entering the soil or emerging from the soil. It's probably going to be the way to go, but Amy's on that one.
Cool, thanks, David.
Yeah, for us it'll be interesting because we've got obviously very dry conditions during the summer, which is not ideal for entomopathogenic fungi. But then there's the opportunity potentially to target mummies on the ground in the winter where there is humidity, and it'd be easier to apply it to them instead of in the tree. So got a couple different avenues we can try to take to use those products.
Yep, and I think that's where it'll head. It'll be looking at trying to get the mature larvae as they're entering the soil to pupate, or the freshly emerged adults as they come out of the soil when they're probably more susceptible. And Amy is doing bioassays to look at the relative susceptibility of those different stages, because it makes sense to target the stage which is very susceptible. Adult beetles are notoriously resistant to a lot of things. They're built in armor, basically. So it's actually hard to tackle them, but all of those different aspects are part of Amy's work.
Just give you a little bit about our sampling on the mummy nuts. So we started sampling in these orchard mummy nuts, and pretty much we're continuing weekly basis, but we start seeing around January, February through March, we're seeing a lot of adults overwintering on the mummy nuts. And around mid-March or so, that's where we start seeing 1 or 2 eggs on those mummies. And then after that, we start seeing the larvae. And that continue, and after time, then after that, we didn't see anything. So I think it's a more like a pupal state. You always see the 1 or 2 larvae though throughout the season, some reason, I don't know. And so around June 1st, I would say June 6th, I think there was a date, we start seeing the adult emergence a little higher number, and then after that it's kind of continuing from that point, which kind of matches with what you have seen in there too, in terms of timing, hull split timing, and their emergence timing too. I think these are all the beetles adults that are active in laying eggs whenever hull split. So it's just the one season we're just exploring all of these things, but these things are very, very difficult to deal with. They have a not very synchronized various things, but and a lot big number in the orchard. So that is critical thing to note.
Yeah, and talking of synchronicity, given that at good times of year they have a generation time of maybe 2.5 weeks, it wouldn't take too many generations to have a complete mix of everything all the time.
Yeah.
After when spring starts firing, you'll get eggs, larvae, young larvae, mature larvae, pupae, adults. If you look, you'll find them there present most of the season, I think.
Hey, really quick question. So you said nonpareils off the tree by September 1st. Is that 2 generations from hull split? Like, is that, if you think of the data, nonpareil splits 2.5 weeks plus 2.5 weeks, is that September 1st? How's that work for you timeline wise?
I would say so, David. Like, as well, certainly from our the numbers of the numbers, that's the number of days and weeks. We haven't actually, I guess, monitored that well. We are looking forward to that monitoring trap from David. So his data, David, you will speak next, but is predominantly lab based, I understand. But your assessment would be reasonable, I would assume.
Yeah, I mean, if even more than that could be possible, right? Because right now we're seeing the beetle laying eggs in the hull split nut. So we're early July. I mean, so early August and early September, we have 2-month period. So, at least the 6 week or 7 weeks. So that's definitely looks like it. If they continue to keep recycling, it's 2 generations, even before that September.
Even before Monterey's come off.
Yeah.
That's scary.
Right. Yeah.
So whatever you do lots of monitoring this season, because this will be the first season you'll actually be able to see what it's actually doing before, during, and after harvest.
Monitoring?
Oh, as in like damage levels, for example, just nut sampling.
Well, it's the first year we're just trying to see how much information we can generate. That's all we're doing right now, because we're focusing on the mummy nuts earlier. Now we're kind of reducing the number of the mummy nut sampling because we kind of get the idea. Now we're increasing the number of the hull split nut sample so that we can inspect the egg and larva in there. But it's challenging because if you bring it in the lab, by then egg might be already fall off and things. But of course, if there is larva, then we'll be able to see it. So that's one of my questions. Actually, earlier today, I was in that orchard, and we're trying to collect the nut from the tree and see how many nuts would be okay or appropriate, at least for that, in order to get a sense of the infestation. 100 nuts, 200 nuts per orchard, because we're very early stage, so I don't think that there is much activity happening yet, but there might be very minimal, but as you go later, we'll get more.
Yeah. A couple of things there. When we first started working in earnest on the beetle, and we collected from solid green unsplit nuts right through the whole split period to see when they got in there. And yeah, they get in as soon as the split's there. But we were collecting carefully. We were basically sneaking up on the nuts on the tree and grabbing them, putting them in a Ziploc bag and sealing it. And we ended up, I think we were collecting 50 nut samples, but from multiple positions on trees, on a set of trees that we're following during the season. And even before we got them to the car, and certainly before we got them to the lab, some of these bags, maybe with, I can't remember if there were 10 or 20 nut samples in a bag, beetles all over the place in there. So yeah, you can't afford to kind of pick nuts and carry them around and do that sort of stuff. You basically need to get them off the tree and bag them straight away to see, just so you don't lose anything. And some of those samples that we're doing And I can't remember exactly what stage they were at as far as split goes. But we used a washing technique where we basically soaked them for a while to get, and most of the beetles and larvae came out, and sometimes you have 15, 20 larvae because they didn't like getting wet and just all coming out through the hull split. As far as numbers go, it's hard. If you want to kind of work out what's going on in tree in detail, then like I said, we collected, I think, 6 samples from a tree, maybe 20 nuts in each of those samples. We did multiple trees. That was really following hull split in detail. If you want to work out where they are in an orchard, it depends on how much time you've got. Because our sampling with our field trials and so on tends to be thousands of nuts, because we want to get a really fine tune the detail of infestation level or distribution. If you want to find out roughly across an orchard, you'd want to be collecting, I think, hundreds of nuts at this stage, hundreds of nuts in a block, across a block, and doing that on all the blocks that you want to survey. If you get just a few nuts in any particular block, it's very easy to miss if there's just a new hotspot. So a block could be infested, but it might only be in one corner if it hasn't actually spread right through the block yet. So there's a risk of missing that unless you get a good scatter of nuts across an orchard block. And how big are the blocks? So we don't have a set number of note sample X nuts in an orchard. I think Ben and Co take 100 nuts just at kind of a random position through an orchard block, which are 15 to 30 hectares, Ben Brown. Yeah. And they get kind of reasonable level of data there, but there's still a chance of missing because it still leaves a lot of trees unchecked. But it comes down to practicality and cost, and how much time and money can you spend as a grower collecting nuts across an orchard to see whether the beetle's there. And that's where the trap is going to come in handy, because you won't have to do all that walking. You'll be able to put a few traps out and see whether the beetle's there or not.
Yeah, thank you. Hopefully the trap will be available soon to use. Yeah. Okay, we're over time, so I don't.
Yeah, we still have a couple of attendees on, and if the panelists, if you need to leave, let me know. We can cut this off at any time. I'll just keep asking some more questions that are popping up. So someone said we had a scenario last season where an almond/walnut grower had 70% damage from the beetle in their almonds, but the walnuts were clean from damage. We believe the walnuts made the situation worse as they are interplanted. So that's more of a comment.
It's worth commenting or replying to, though. And this, again, these 3 pests, navel orangeworm, carrot moth, and Carpophilus, are quite similar in a way. You can get quite high levels of infestation with carrot moth, at least between the hull and the shell, and no kernel damage. And particularly, like if you have hard shell varieties, you may find that they're still fine for carrot moth, say, to breed in and to spread out and infest susceptible varieties. I imagine it'll be the same with Carpophilus beetle, where they may fly around and lay eggs in hull split nuts. They may never get into the kernel on those nuts, but they still feed on the outside of the shell and the inside of the kernel to the point where they can reproduce and then spread to susceptible varieties. So it could be in that case that if that walnut was a really hard shell one, it's possible that the beetle was breeding up in the walnuts and then spreading to almonds where it actually did the damage. But without knowing more detail, they would have to have sampled those walnuts during the beetle reproduction kind of period to see if they were actually using them or not, or whether that's a furphy and they actually came from somewhere else. But that's certainly a possibility.
It would be interesting if that person told us what variety of walnuts, because as Jalindra said, a Chandler might be totally different than another variety just because there are hard and soft shelled varieties. So maybe it's just a walnut that's a good variety, and if it had been a different variety, maybe it would have been an opposite result. And just like you said, David, that more details are needed there to really figure out what's going on. Yeah.
And it reminds me of when carrot moth really fired up kind of years ago, some growers are going out and just spraying the nonpareil rows because that's what's being damaged, but the carrot moth would have been breeding up in every second row, which is a pollinator. Just kind of defeated the purpose.
Yeah, that was an anonymous comment. So whoever said that, if you want to share the variety, go for it. Next question, are the beetles killed by freezing post-harvest? I assume pasteurization kills them at least.
The beetles don't like cold, as in when we put samples in the cool room, the adults will die relatively quickly. And then when we get around to assessing those nuts, we can still find live larvae. Not after weeks or anything like that, but certainly after maybe a week or two. If you freeze them, everything will die. Pasteurization, I'm not sure of the process of how much that actually heats the entire nut as opposed to just the surface. So someone who knows the pasteurization process might be able to answer that. If it basically scalds the surface and then it chills again and the internal of the nut doesn't get hot, they could quite well survive, I imagine. Don't know.
Ben Wiblin, were you gonna say something? Or
I probably don't really have anything too definitive there, but we do pasteurize, but not 100% of the crop, just for export markets that require it, or more so, sorry, the domestic market. But of course, like in our post or processing or finished goods warehouses, all storage controlled in terms of temperature, just to slow or stop any life cycle that could be building up or whatever in that room.
So Ben Wiblin, do you have any idea of the internal temperature of nuts when they're pasteurized?
Yeah, that's the answer I didn't have for you, David, so I didn't want to jump in too quickly. But yeah, I think it's obviously a pretty strict heat treatment, whether or not the penetration within the kernel. I'm not sure the internal temperature that it reaches to answer that question properly. Probably so, David.
Okay.
Yeah, and as for freezing, of course, freezing kills them. But whether there's a table that shows how long you have to freeze it to kill each specific stage, I don't know if those tables exist.
Not yet. There's a job for you.
There you go. Jalendra will do it.
There is another question about economic threshold for insecticide use, but I think you mentioned already, David, Madge, that you don't really have an economic threshold at this point, correct?
We don't have any economic threshold for mummy nuts like you guys have developed for navel orangeworm. So no, that's one for growers to, I mean, I guess it comes from experience, is basically, and it'll only come from a lot of monitoring data and experience of, no, if we're looking at, we're going to end up with 10% damage, is it worth putting a spray on? Given that we know that sprays are very hard to get in the right place, and grower anecdotes that I hear is that we don't see great benefit from them, at least as a one-off spray. So I'd say, no, we don't have a threshold for putting a spray on.
The only other thing I'd add to chemical controls, I think we hinted at it before, or at least Ben Brown did in his presentation, is uniformity of split. So we're talking about the stage of application of chemical, and when the Carpophilus beetle gets in, and you're looking at 5% hull split, right? But not all your tree is at 5% hull split at that stage, let alone within your block, within your farm. So that decision about when to actually apply that, and then you got to deal with residues at the end. So when your majority of your nuts are at 5%, then your other ones are almost split. Like, what's the effect then on your residues? It's a whole, I guess, thing where you have to consider. Obviously, if we can even up our production in terms of that split, it'll have benefits for harvest, for hygiene, for everything. So for quality, yeah, it plays a big role, I think.
Yeah, so insecticides from a California perspective. So our standard practice is an application at the start of hull split, a second application 2 weeks later for navel orangeworm with methoxyfenazide and chlorantraniliprole. Bottom line is that program is going to do nothing for Carpophilus beetle. So then if you look at bifenthrin, so we have bifenthrin and lambda-cyhalothrin. That's our two primary pyrethroids. Some people do add those to those sprays. They're going to do something. The question is whether or not the help they give on Carpophilus or the help they give on navel orangeworm, which in some cases is resistant to them, offsets the harm you cause by flaring mites. So that's the balance. There's no thresholds. There's no perfect answer for that. That's what a grower has to consider with those. Tetraniliprole, we don't have registered in California that I'm aware of. That's another diamide. And then the clothianidin, that's not something we currently use. That's belay, for those of you not familiar. That's not something we currently use in almonds, other than maybe sometimes for stink bugs or for leaf-footed bugs. but it's certainly not a product we're used to using at hull split or post hull split, which would mean an extra, is it worth, are you getting another, I don't recall what it costs, but are you getting another $40 per acre, let's say, of benefit out of adding that product in as a tank mixed with the navel orange worm spray? Again, we don't have an answer to that question. But that's kind of where we are, and people are just going to have to juggle those pros and cons to decide if they're going to try a chemical control program.
Yeah, basically this year we don't have any data, right? So you can't say anything. It's just to make an assumption of some of the things, but you can't say definitely. Hopefully, and the navel orange trial or Carpophilus trial looks like similar thing. Hopefully, we'll set up the trial this week. We're going to go that first hull split spray with the multiple insecticide on that targeting Carpophilus and both navel orangeum. And we're doing this replicated trial and see what we'll have at the end of the season, the result wise. We'll, try to use the insecticide that we're already using and see whether we can see the effectiveness on this Carpophilus beetle. But again, this season, it's just based on everyone kind of like, experience and assumption that it might work. That's where we are now. Yeah.
Yeah, that's the status of the information, and use it to make whatever decisions you feel are appropriate for your particular situation.
Yeah. So it'd be very interesting to see how that spray trial goes. With the IPM, the 2 IPM projects we've run, or we're running the second phase of it, we were explicitly, don't know how to say it, required not to do any chemical spray trials. If the industry decides that they want a spray trial, they need to approach the funding body who funds our projects and get a particular trial set up to do that. It was definitely kind of excluded from our scope. Luckily, from my point of view.
All right, I think maybe we can wrap up with this question. Ben Brown, you addressed it in the chat, but the question / comment was, I appreciate the discussion and all the wealth of information, which it has been great. For California PCAs and growers, what is if there's one thing to remember today, message? In other words, we're in mid-hull split. What should we be doing right now? And what's the one thing to take away from today?
We kind of answered that. For the second part, we kind of answered that. The first part,
harvest as soon as possible, and then sanitation.
Yeah, exactly. The first part is sanitation, because I mean that's been my experience. When you go these orchard with a lot of mummies on the ground, every one of them you open up, you see the beetles, you see the egg, you see the 10-15 larvae inside those mummy nuts. There is no other places that I've seen the beetle on the ground. They're not crawling around, nothing. It's just all on the mummy nut. So what's in there, and that's where we're seeing that these are coming from there and going up in the tree. So that's seems to be a bottom line.
And if you're sweeping all of those infested mummy nuts up with your new crop that you've just dropped on the ground, fumigation as soon as possible is really, really critical.
Yeah, sanitation, the final word. All right, so I think we can wrap up with that. I really appreciate all of you being here. I appreciate all the attendees, especially the people who are still sticking around right now at 6:30. I think they're here because they care and because you all have shared some really incredible information. There were questions in the beginning, as I kind of alluded to. Once this recording is processed and we put it on a website, I'll share with everyone and let them know where it's at. Yeah, if you have questions about DPR stuff, email me. My email address is in the chat. And you should have gotten an email from me about the webinar, so you'll be able to find me easily. But yeah, one last huge, incredible thank you to the Australians and to our California researchers. So yeah, everyone have a good evening and good luck this season.
Thank you,
Thanks, Katie, for
Yeah.
hosting it.
And good luck.
Thank you.
Thanks, everyone.
Yeah.
Thank you so
Thanks,
much.
everyone.
Really appreciate all the contribution. It was really useful information that we gained from from your experience. So I appreciate it.
Yeah. And I'm sure we'll all see much more of each other in the coming months and years.
Seems like it.
Thank,
All right. Yeah,
Thanks, everyone.
thank you all. Have a
Thank
good
you.
Bye.
day in Australia.
Bye.
Good evening, California.
Bye.
Bye.